Okay, you did revision. Just have you go through something. We'll just have small revision, whatever we have done yesterday. Enzymes and carbohydrate metabolism. Okay. So in enzyme inhibitors, statins inhibiting HMG-CoA reductase. So how about the Km and Vmax in this condition? Statin inhibiting HMG-CoA reductase is an example for an inhibitor. How about Km and Vmax in this condition? First find out what is this inhibition. Nothing is here. Okay, just go through your notes. What type of inhibition? Statin inhibiting HMG-CoA reductase. Competitive inhibition, yes. So in competitive inhibition, how about the Km and Vmax? Vmax remains same. Km increases, yes. Okay. So alpha ketoglutarate. Okay, give one example for non-competitive inhibition. Give one example for non-competitive inhibition. Sodium fluoride inhibiting enolase, anything else? Alpha ketoglutarate inhibited by what? See and tell me. Yes. Have you written or not? Written? So tell me. In glycolysis, glyceraldehyde 3-phosphate dehydrogenase is a intermediate step in glycolysis, which is inhibited by arsenate and iodoacetate, and also in electron transport chain, complex 4 inhibitor, cyanide, carbon monoxide, hydrogen sulfide, sodium acide. All those inhibiting the complex 4 of electron transport chain are examples for non-competitive inhibitor. So can you tell me the classification of enzymes? 1, 2, 3, 4, 5, 6, 7 in order. Oxidoreductase, yes. Transferase, hydrolase, lyase, isomerase, ligase, translocase. Okay. So synthase is an example for. Synthase will come under which classification? Synthase, ligase, synthetase, synthetase is ligase. Synthase will come under lyase. Glycogen synthase, transferase. Okay. Sodium potassium ATPase is an example for transferase or translocase? Translocase. Okay. Enolase is an example for lyase. Yes. Okay. Which of the following metal is an metalloenzyme for lysyl oxidase? Lysyl oxidase. Copper. Superoxide dismutase. What is the metal? Copper. Xanthine oxidase. Okay. Alcohol dehydrogenase. Alkaline phosphatase. Lipase. Urease. So what is the enzyme deficiency in 1 Greek's disease? What is the enzyme deficiency in von Greek's disease? It is a glycogen storage disorder. So what is getting stored? Glucose 6 phosphatase. What is that enzyme deficiency in McArdle's disease? McArdle's disease. Muscle phosphorylase. What is the enzyme deficiency in Hirsch's disease? Okay. What is the enzyme during glycogenolysis? During glycogenolysis, you know what is glycogenolysis? Which of the following enzyme is absent in muscle? Hence, muscle cannot contribute for maintaining blood glucose. It can contribute only for energy. What is the enzyme absent in muscle? It's glucose 6 phosphatase. Okay, so what is the irreversible step of glycolysis? 3. Irreversible step. 3 important I said for glycolysis and gluconeogenesis. Pyruvate to acetyl-CoA? No. Glycolysis means it ends with pyruvate or it ends with lactate. Conversion of pyruvate to acetyl-CoA will not come under glycolysis. It is a link reaction between TCA and glycolysis. What are the 3 irreversible steps of glycolysis? Glucose to glucose is phosphate. Tell me only the enzymes. Hexokinase, phosphofructokinase, pyruvate kinase. So what are the 4 important enzymes, irreversible enzymes in gluconeogenesis? If it is gluconeogenesis, you should start from pyruvate. Pyruvate, yes. Before that, pyruvate carboxylase. The next one is phosphoenol pyruvate carboxykinase, PepCK, and then fructose 1,6 bisphosphatase and glucose 6 phosphatase. So how much is the amount of ATP generated from one glucose after complete oxidation? 32. Complete oxidation means 32. Can you tell me how it is 32? First is glycolysis. How much? 7. Then, so at the end of glycolysis, you will get how many pyruvates? 2 pyruvate. So that 2 pyruvate will be converted to 2 acetyl-CoA. So how much energy produced in link reaction? 5. 2.5 into 2. And then 2 acetyl-CoA entering TCA cycle. So 2 into 10. 1 TCA cycle will give you 10 ATPs. How 10 ATP you know? 3 NADH step, 1 FADH step and 1 GTP step. So 20. 20 plus 5 plus 7. So you will get 32 ATP. So how much ATP produced from anaerobic glycolysis? 2. Clear? Okay. So what is the rate limiting enzyme for glycogen synthesis? What is the rate limiting enzyme? Even if you don't know any enzyme, no problem. This enzyme you should know for glycogen synthesis. What is the rate limiting enzyme? Glycogen synthase. Okay, what is the rate limiting enzyme for glycogenolysis? Glycogen phosphorylase. Yes? Okay. So which of the enzyme will be active in dephosphorylated state? Out of these 2 enzymes, which enzyme will be active in dephosphorylated state? So if I ask like this, which enzyme active in dephosphorylated state, first thing you should come to your mind is which is activated by insulin. Yes. So insulin will be activated which enzyme? That you should know which is happening in fed state, which is happening in fasting state. Insulin activate the pathways happening in fed state. So which is happening in fed state out of these 2? Synthesis. Glycogen synthesis is happening in fed state. So the enzyme glycogen synthase will be active in dephosphorylated state because that will be activated by insulin. So which enzyme will be active in dephosphorylated state? Sorry, which enzyme is active in phosphorylated state? Glycogen phosphorylase. Okay. So what is the enzyme deficiency in classical galactosemia? Classical galactosemia, the main enzyme deficiency. Go to galactose metabolism. Galactose 1-phosphatase uridyl transferase. That is for classical. Okay. So why there is cataract in galactosemia? It is called oil drop cataract. The name of the cataract is oil drop cataract. It's like some bubble, water bubble or oil bubble. So why there is cataract? Which enzyme is responsible for causing cataract in galactosemia? Aldose reductase. Okay. So what is the enzyme deficiency in hereditary fructose intolerance? Aldolase B. So aldolase C will be present in brain. Okay. So which of the following enzyme deficiency is the most common cause for hemolytic anemia? That patient is resistant to malarial infection. He has not attained infection anytime in his life. So G6PD, glucose 6-phosphate dehydrogenase. So this enzyme will come in which pathway? G6PD enzyme will be seen in which pathway? Glycolysis? No. It will come from one oxidative pathway. What is that? HMP pathway. It is the first enzyme in HMP pathway. Glucose 6-phosphate dehydrogenase. What is the second common enzyme responsible for hemolytic anemia? Pyruvate kinase. So why there is RBC affecting in this condition? Why it is always first RBC to affect in this condition? Why not other organs? Because lack of mitochondria, no other energy pathway can happen in RBC. It has to depend only on glycolysis for glucose and HMP pathway. Okay. So what is the enzyme deficiency in essential pentosuria? Essential pentosuria. So only 3 points I have said in that. For which pathway? If it is essential pentosuria, patient is harmless, no other complaints, only reducing sugars positive in urine. What is the enzyme deficiency? Xylitol, xylulose, reductase. Second is xylitol dehydrogenase. Okay. So what is Gerard's tetrad? Which 3-4 inborn errors will come under Gerard's tetrad? Cystinuria, albinism, alkaptonuria, pentosuria. Okay. Okay. Can you name some sphingolipidosis or glycospingolipidosis? Each one name of the disease. Von Wig's, the most important thing is, if I tell Von Wig's, you should not confuse whether it is lipid storage disorder or glycogen storage disorder. You should be very careful with that point. The first 7, Von Wig's, Pompey's, Cory's, Anderson's, Hers, McCardell's, everything is glycogen storage. When it comes to lipid storage, it starts from GM3, GM1, GM2, GM3 gangliosidosis, sialidosis, Fabry's, Gaucher's, and then Farber's. So all these are lipid storage disorders. Okay. So what is enzyme deficiency in Tay-Sachs disease? Hexosaminidase A, Sandox. Hexosaminidase B, yes. Okay. What is the important clinical feature in Fabry's disease? Fabry's, all patients will present with mental retardation, corneal clouding. But in Fabry's, what is that important thing? Red purple spots. Okay. In which of the glycosphingolipidosis, there is no corneal clouding. No corneal clouding. No corneal clouding. So I said one way to remember it easily, Hunters need eyes. So there is no corneal clouding in hunters. So which of the following glycolipids, lipidosis is X-linked recessive? All are autosomal recessive except one. What is that one? Hunter's disease. Okay. So we'll start. So we'll continue with lipoproteins, what all the types of lipoproteins, and then we'll go to metabolism like fatty acid synthesis, beta oxidation, what is happening. Okay. So what is lipoproteins? From the name, what you can understood this lipoprotein. So for what we discussed is only lipids. Yes, but here it is lipid with proteins. Yes or no? Okay. So this is the structure of lipoprotein. So this is the structure of lipoprotein. This blue color, this is the outer shell. So inside it is that core lipid. So this outer part is made up of unesterified cholesterols. So this is looking like this. Can you see like this phospholipid? So like this, it will form a outer membrane. So inside our triglycerols, triacylglycerols, and our cholesterol will be condensed inside, which is covered by our proteins with unesterified cholesterols. So this protein, which is responsible for identifying what type of lipoprotein, is called apoprotein. Okay, so along with this peripheral lipids, phospholipid, you will have an apoprotein. So what is the function of this apoprotein? Since it carries apoprotein, it is called as lipoprotein. Understood? What is that protein part in lipoprotein means? It is apoproteins. So what is the function of this apoprotein means? There are different types of lipoproteins. One will carry chylomicron, triacylglycerol. One will carry LDL. One will carry VLDL. One will carry good cholesterol. So depending on that, we have to separate which is carrying which lipids. So that will be identified by different types of apoprotein present on its membrane. Okay. So what are the types of lipoproteins you know? You know chylomicron. Have you heard of chylomicrons? Have you heard VLDL? Very low density lipoprotein. Very low density lipoprotein. Next is low density lipoprotein. Low density lipoprotein. Next is IDL, intermediate density lipoprotein. Next is high density lipoprotein. This is high density lipoprotein. VLDL, LDL. First is chylomicrons, VLDL, LDL, IDL, HDL. So all these five together we are calling as a common term. They are lipoproteins. Okay. So how it is classified? Something is called chylomicron, something is called VLDL. So each and every lipoprotein is having some characteristic properties. So what is that property? So just see here, which one is looking very big? Is it looking big? Only half has been covered. So this is the bigger one. Yes. So the next one is VLDL. Next one is IDL. Next one is LDL. And the next one is very smaller one is HDL. So depending on size, it is increasing in this way. Is it right? HDL is small, chylomicron is higher. So as per the size, it is increasing from HDL, LDL, IDL, VLDL and chylomicron. Yes? Okay. This is size. Now I am going to tell about density. Density means which is more dense, which is even though size is big, it will not occupy that much. Even though size is small, it can occupy so much. So this is depending on size, it is increasing in this way. If it comes to density, which is more weighted one, it will go in this way. Since because size is bigger, it does not mean the weight is more. HDL is having more high dense lipids comparing to all other food. Okay. Clear? Okay. So next is out of this 5, which is having more lipids, which is having more of lipid content. It is triacylglycerol and cholesterol, which is having more of lipid content. Means chylomicron is having more of lipid content. So your lipid content will be higher in this way. Your lipid content is higher in this way. It means HDL is having very less fat, lipids, triacylglycerol and cholesterol. Whereas chylomicron is having high amount of triacylglycerol and cholesterol. So then you can have a doubt, when chylomicron is having high lipids, how come density is less? Because HDL is having more of protein content comparing to lipid content. So for occupying a lipid, we need space, but weight will be less. But to occupy a protein, we need less space, but weight will be more. I am repeating again. So this is chylomicron. Size-wise, it is bigger. Lipid content-wise, it is bigger, but density-wise smaller because it is having only lipids, very less amount of proteins. So your lipid weight will be less. But when you come to HDL, lipid content is very less here. Lipid is very, very less. Protein content is more. So to occupy a protein, we need less space, but it will give you more weight. That's why the density is increasing with HDL. Even though size is small, it is having protein. So the weight is increasing in HDL. So when it comes from chylomicron, VLDL, IDL, LDL and HDL, slowly it is removing the lipids and taking up the proteins. It will remove lipids and take up proteins. One by one, same thing is happening. So finally, when it comes to HDL, there is very less lipid content, but very high amount of protein. That's why even with small size, it can occupy large amount of protein. It is giving the weight for HDL. That's why the name itself is high density lipoprotein. Chylomicron, next is very low density. Next is high density lipoprotein. So from the name itself, you can tell which is more dense. The reason is more of protein content comparing to fat content. Okay. So in this whichever is having yellow color, yellow resembling the fat, all are bad cholesterols. Whereas which is having more of protein content is good cholesterol. Okay. So before going to this, okay. So I will take lipoprotein, which is chylomicron, VLDL, LDL and HDL. I am not including IDL because IDL is like a link between your VLDL and HDL. VLDL will be converted to LDL. In between, we have IDL. So this IDL is resembling like VLDL and LDL. So I am not going to include IDL in my explanation. It will come, but it does not have any separate properties. It is going to be converting the VLDL to LDL. In between, your ideal intermediate density lipoprotein will come. So each and every lipoprotein is having a function. Okay, so we are having food. We are taking food. Yes, we are taking food. So it is reaching intestine. Yes or no? Before going to blood, everything, it is reaching intestine. So from the intestine, some of this lipoprotein has to carry our lipid content to where? To blood, from blood to liver for metabolism. Yes or no? Everything has to go to liver for metabolism finally. So what we have taken outside from the food will reach the intestine first. From the intestine through blood, it will be taken to liver. So that function is carried by chylomicrons. So what is the function of chylomicron is? It carry exogenous fat. Exogenous means from outside, whatever we are eating. Carry exogenous fat from intestine to liver for metabolism. Okay, clear? This is the function of chylomicron. So next is VLDL. What is the function of VLDL? So where it is synthesized, where chylomicron is synthesized, it is synthesized in intestine because the function starts in intestine. So it is synthesized in intestine. So next is VLDL. The next lipoprotein VLDL. What is the function of VLDL? One lipoprotein has carried whatever we have taken to the liver for metabolism. Now the function of VLDL is to carry triacyl glycerols. To carry endogenous triacyl glycerol. What is meant by endogenous triacyl glycerol? Fatty acid synthesis happens. Yes or no? In liver, fatty acid synthesis will happen. So after that, we will end up with fatty acids. So that fatty acid has to go somewhere else and store. If we are not synthesizing, it's good. If we are synthesizing more fatty acids, if it gets deposited in liver first, it will go for fatty liver. To prevent that, what our body will do, it will carry that fatty acid from the liver to store somewhere else in our peripheral tissues. So the one which is dedicated for storing fat is called adipose cells. Yes or no? So the function of VLDL is carrying endogenous fat. Endogenous fat means whatever has been synthesized in our body itself. Carry endogenous fat from liver to adipocytes. So adipose tissue is for storing of fat. So once fat has been synthesized in liver, it has to be taken to adipose tissues. So that function is carried by VLDL. It is synthesized in liver. The function starts in liver, it is synthesized in liver. Okay. In endogenous fat, I will mention it as triacyl glycerol. Mainly it is carrying triacyl glycerol. VLDL is having more amount of triacyl glycerol. Yes. So next is LDL. What is the function of LDL? LDL will be carrying the cholesterol from the blood to periphery tissues and also to the liver back. It is carrying cholesterol. This is carrying cholesterol because LDL will have high amount of cholesterol compared to triacylglycerols. So carries the cholesterol from blood to liver. Why it has to take the cholesterol back to liver? Any idea why it has to take the cholesterol? Now only VLDL has carried the triacylglycerol and stored in adipose tissue. But now this LDL is carrying back the cholesterol back to liver because cholesterol is essential for synthesis of steroids, bile salts, and for all fat soluble vitamin to be absorbed, we need cholesterol. So cholesterol itself is having function in our body. So it has to go back to liver. So that is LDL. So next is HDL. What is the function of HDL? The function of HDL is it carries fat, that is triacylglycerol, from peripheral tissues back to liver. from peripheral tissues back to liver. See, all these are carrying exogenous fat or endogenous fat from intestine to liver, from liver to peripheral tissues. Whatever LDL has done to us will be reversed back by HDL. So why it has to be reversed? Storing of fat is not good. Yes or no? Storing of fat in peripheral tissues, not only adipose tissue, it will store in blood membranes also, in blood cells, in blood vessels also it will store. Peripheral tissues means other organs, adipose tissues, blood vessels. If your triacylglycerol starts to accumulate in blood vessels, it will cause atherosclerosis. So to clear those fat which has been stored in blood vessels and other organs, we need HDL. So now this HDL will take the cholesterol. I will write fat. That is especially triacylglycerol will be very less. Cholesterol is also very less in HDL. It is having more of protein content. So triacylglycerol and cholesterol from peripheral tissues back to liver. So now I will draw a diagram so I think you can understand. So this is intestine. This is intestine. Okay. So whatever food we had is having what fat? Exogenous fat. This is intestine. So whatever we ate, that is coming to intestine. So this is carried from intestine, blood, and then it reaches liver. Okay. So this is done by chylomicrons. So next is VLDL. What is the function of VLDL? It carries from liver to peripheral tissues. Now I will make blood here. Peripheral tissues, I will make an adipose cells. This is adipose tissue. Okay. So from liver, this fat is carried. Who is carrying this fat? VLDL. So VLDL is a lipoprotein. Okay. So this is carried as VLDL from liver to blood. It reaches the blood. So how it reaches the blood? So here comes that protein part. I said lipoprotein. The lipid part is triacylglycerol. Then what is that protein part? What is it called? Apoprotein. I said or not? Apoprotein. So this apoprotein present in VLDL is ApoB100. ApoB100. So there are different apoproteins. To carry from intestine to liver, we need chylomicron. So that chylomicron is having B48. B48 means apoprotein B48. That is the protein present in that chylomicron that will do the function of carrying from intestine to liver. So next is VLDL and LDL. Both are having B100. That is a clue for VLDL and LDL doing the function of carrying from liver to periphery. Next comes HDL. HDL is having apoprotein A1. A1 is the protein present in HDL. So this is specific for chylomicron, VLDL and HDL. So apart from this, it also carries ApoC2, ApoE. ApoC2 and ApoE. If I make this and if I give you, this is B48, C2 and ApoE. So now I will ask you, what is this lipoprotein? What is this lipoprotein? Having ApoB48, C2 and E. Okay. So now tell me, what is this first one? What is this first lipoprotein? Having ApoB48 as apoprotein, C2 and E on its surface with 99% of triacylglycerol. So what is this lipoprotein? This is chylomicrons. Clear? So how I can tell it is chylomicron? Two things. It is having high percentage of triacylglycerol. But apart from that, the most important is the lipoprotein, which is coding for chylomicron. It is ApoB48. So now what is this second one, which is having triacylglycerol and also cholesterol with ApoB100, ApoC2 and ApoE. So don't see 3 and 4 pictures. Don't see this one and this one. If you see only 2, you can tell it as either VLDL or LDL. Both are having ApoB100. Yes. But when it comparing 2 and 3, you can tell triglycerol content is reduced, cholesterol content has been increased. So comparing to VLDL and LDL, VLDL will have more of triacylglycerol compared to cholesterol. But LDL will have more of cholesterol compared to triacylglycerol. See, here it is, triacylglycerol is more comparing to cholesterol in VLDL. In LDL, cholesterol will be usually more, 60% I will make. So cholesterol will be more than triacylglycerol. Okay, so in this two condition, you can tell this is VLDL and this is LDL. So that's why in conditions like atherosclerosis, increased blood LDL level is very bad for atherosclerosis, because this cholesterol, it is carrying the cholesterol from where it is carrying. LDL is carrying cholesterol from liver to periphery. Periphery means adipocytes or blood vessels anywhere. So if the cholesterol goes and accumulate in blood vessels, it can cause atherosclerosis, myocardial infarction. So that's why LDL is more bad. But when VLDL is high, it means triglycerol is high. So when triglycerol is high, comparing to atherosclerosis, patient is having more risk of getting diabetes. Is it true? Can diabetes, can blood sugar can increase triacylglycerol level? Yes. Why? How? Glycolysis, glucose. What is the intermediate step? Glyceraldehyde. Yes or no? Glyceraldehyde 3-phosphate. So now this glyceraldehyde 3-phosphate, already we have taken more glucose. That's why I am talking about diabetes. Already we have more glucose which went to pyruvate TCA cycle energy. So now what this glyceraldehyde will do? It will divert to formation of glycerol. So now this glycerol, if free glycerol has been formed from glycerol 3-phosphate, this will combine with whatever less amount of fat we have taken, and this will form triacylglycerol. That's why when we increase our glucose intake also results in fat accumulation. That is triacylglycerol accumulation. So that is the reason when we check our lipid profile, If triacylglycerol is very high, first we are going to get diabetes. Next, we are going to get all diabetic complications. Clear? If LDL is high, it means the direct cholesterol itself is going and accumulating in the peripheral tissues. So it is more risk for atherosclerosis. Clear? But triacylglycerol is high, it means the more risk is for diabetes compared to atherosclerosis or myocardial infarction. So when you see here in VLDL, triacylglycerol content will be more. In LDL, cholesterol content will be more. Both are having same apoproteins. Clear? So next comes HDL. HDL, first of all, the size will be smaller comparing to all other, and then it is coded by the apoprotein A1. It is going to carry whatever has been stored in the periphery. It will collect everything and give it back to liver for excretion. That's why it is called good cholesterol. HDL, high density lipoprotein, is also called good cholesterol. When we have more HDL in our body, it will carry more amount of cholesterol from the periphery to the liver for excretion. If our HDL is less, LDL is high. What is happening? See, HDL is less. My LDL is very high. So what my LDL will do? It carry from liver to blood vessels and start accumulating cholesterol here. Yes. If my HDL is not there, it cannot go back. If my HDL is there, if HDL is present, this will be removed from blood vessel and goes to liver for excretion. If HDL is not there, LDL is high, it will start to accumulate in blood vessels. So this is very, very important topic in clinical point. Lipid profiles, all kind of dyslipidemias, many hyperlipoproteinemia, many clinical conditions can be seen with this. Okay, now you understood what are that for? Chylomicron, VLDL, LDL, HDL. Only if you know the function, you can apply it. You know the functions, right? Chylomicron, VLDL, LDL and HDL. Okay. So I was telling something here. So this is having triacylglycerol. Okay. More of triacylglycerol. So then what is this lipoprotein? If it is carrying more of triacylglycerol, then what is this lipoprotein? B48. 100 or 48? 100, because only from intestine to liver it is chylomicron B48. So now here it is ApoB100. So this ApoB100 carry this triacylglycerol to blood vessel. So now this triacylglycerol, which is present in blood vessel, will be activated by ApoC2. We have ApoC2 also. Yes or no? I said it will have B100, ApoC2 and ApoE. So now this ApoC2 gets activated by lipoprotein lipase, which is present in blood vessel membrane. Clear? See, this is EC2 with B100. So you know this is carrying triacylglycerol more comparing to cholesterol from liver to blood vessels. So now it is reaching blood vessel. Once it reached blood vessel, it has reached the blood vessel. Okay. Now whatever the C2 is present, it will activate lipoprotein lipase, which is present in our membrane, blood vessel membrane. So what is this function of lipoprotein lipase? This is very, very important. This is very, very important. This is lipoprotein lipase. Lipase means, what is the function of lipase? Cutting. So what it is going to cut? It is going to cut this lipoproteins. It is not going to cut that lipoprotein directly. It is going to cut the content which is in that lipoprotein. So what it is carrying? It is carrying triacylglycerol. So this triacylglycerol will be broken by this lipoprotein lipase into glycerol and fatty acid. You know, triacylglycerol is made up of 1 glycerol and 3 fatty acids. So this triacylglycerol has to be cleaved into glycerol and 3 fatty acid by an enzyme called lipoprotein lipase, which is present in our blood vessels. Okay, so this all happening in blood. So now this fatty acid, which is bad for our blood, has to enter to peripheral tissues, any tissues. First it will go to adipose tissue. If adipose tissue is not there, it will go and deposit in the organs. So this fatty acid will reach us adipose tissue, and here it will again combine with glycerol and stored as triacylglycerol. So the storage form of lipid in our body is triacylglycerol. The storage form of glucose in our body is glycogen. The storage form of lipid in our body is triacylglycerol. Clear? This fatty acid and this glycerol enters to adipose tissue, where it can again combine to form and deposit as triacylglycerol. Yes. So now what happened to this? What happened to this chylomicron? It has given some triacylglycerol. Yes or no? From this triacylglycerol only this has been broken. So now this lipoprotein will become little smaller. Can I write? Because some of the content has went out. So I can make it as little smaller one with same B100, C2 and E. So now this is called IDL. Okay, just smaller than that, with some triacylglycerol has been removed. So now it is called as IDL. Now this IDL will decide whether I should go back to liver for excretion, that is destroying, or I should continue in the blood. Clear? So this IDL, if we are having more amount of free radicals, if we are having more amount of free radicals in our blood, what this IDL will become? This IDL will get that free radical and it will become oxidized LDL. Now this will be converted to LDL. This is LDL. So this LDL, you know, already triacylglycerol has been removed. So now this LDL is having more of cholesterol compared to triacylglycerol. Is it true? Because here TAG has been removed. So now when you compare in LDL, which is more, cholesterol will be more compared to triacylglycerol. So now this LDL will start to get deposit the cholesterol in blood vessels. This LDL will get deposit cholesterol in the blood vessels. Once it has deposited, the final one is called remnant. LDL remnant. Remnant means the waste one. So this LDL remnant has also go back to liver. Okay. So either this IDL will go back or this LDL will go back to liver. So now liver has to take this and it has to degrade it. So how the liver will identify this two? Here comes the function of ApoE. Here comes the function of ApoE. Our liver is having receptor for ApoE. This is called ApoE receptor. So whatever may be the lipoprotein, chylomicron or IDL, LDL, VLDL or HDL, all will have ApoE in its surface. So what is the function of ApoE? This is now LDL with B100, C2 and E. This E will come and join here. So if I make another one, this will come and join here, then only it will take it inside. So all chylomicron, all lipoproteins will have ApoE on its surface. The function of ApoE is it acts as a ligand for ApoE receptor, which is present in the liver. Clear? So what is the function of ApoC2? What is the function of ApoC2? Activating lipoprotein lipase. So function of ApoC2 is activating lipoprotein lipase. Then only from VLDL, triacylglycerol can get broken into glycerol fatty acid. Both can go into adipose tissue, again store it as triacylglycerol. What is the function of ApoE? It is a ligand. It is a ligand for ApoE receptor, which is present in liver. Only if ApoE is there, it can go back to liver for degradation. If ApoE is absent, if our lipoprotein is not having ApoE, it cannot go back to liver. It will be circulating in our blood only. Yes? Clear? Okay. So this is the function of, this is how VLDL converted to IDL and it has converted to LDL. Finally, it has to go back to liver for degradation. If E is absent, it cannot go back. It will circulate in the blood. When it is circulating in blood, we will have more amount of LDL remnant, VLDL remnant. Because the other function is happening, so it will become the remnant. It will remove the cholesterol, triacylglycerol and become remnant. But once the remnant has to go back to liver, will not happen. It will be circulating. Yes. So now, what is the function of HDL? So now the function of HDL is, consider this as some peripheral tissues like muscle. Okay. This is blood. Now here it is liver. So now the HDL, at first it is disc shape. Okay. At first it will looks like a disc. Once it is circulating in blood, this peripheral tissues will tell I have more of excess fat. I have to remove that. So all this cholesterol from the excess fat will be taken by this HDL. And once it has taken more cholesterol, it will become little bulky and it will be circular, spherical shape. Okay. So now this is active HDL. So your HDL is having what all the apoproteins? A1, same C2 and also E. Okay. So once it has taken all this, it will not release the triacylglycerol to blood vessel because HDL is having more of protein content. It will take only whatever the excess fat from peripheral organs. So here your C2 will not have any role because triacylglycerol itself is less. It is carrying from the peripheral tissue to liver. So this A1 and E is recognizing this HDL. Now from the blood, it will reaches the liver. In liver, you know, we have ApoE receptor, which will also take this HDL back. Yes, that's why it is called good cholesterol, because it is carrying from the periphery to liver. Okay, so for this, from peripheral tissues to enter the fat to the lipids, we have some transport proteins. They are called as ABCA1. Just remember, we have another one which is ABCA1, and here we have SRB1. Okay, like similar to ApoE, we have SRB1 that will recognize this HDL also. So remember only these two transporters. One is present in periphery, one is present in the liver. So if ABC1 is the transporter for taking the lipid, fat, triacylglycerol from periphery to blood, if this is not functioning properly, if ABCA1 is not functioning properly from periphery, it cannot enter to HDL. Yes or no? If this is absent from periphery, it cannot enter to HDL. So HDL level will be less. So this condition is called as tangential disease. Tangential disease means on blood lipid profile, patient is having less HDL. It does not mean that HDL production is less. HDL is produced properly, but the function of HDL is not able to carry it out because of transporter of ABCA1 defect. So your HDL is present in this form, which is an inactive form. The proper active spherical HDL has not been formed. So decreased HDL level due to ABCA1 gene defect is called tangential disease. So patient is having more of lipid in the periphery because the clearing is not happening. Okay. So this is that HDL. Here you can see free cholesterol from peripheral tissue has to enter. So this part is called ABCA1 transporter. That will be absent. Okay. Okay. So this LDL receptor. So how that final IDL and LDL will go into liver? What I said? What is taking that remnant LDL back to liver? With ApoE receptors. Yes or no? ApoE receptor for IDL, VLDL, for HDL, and also for chylomicrons. For LDL, apart from that, we have one more clearance activity, that is LDL receptor mediated endocytosis. So this is our liver. This is our liver surface. In this surface, you can see receptor. Similar to ApoE receptor, we also have LDL receptor. So what this LDL receptor will do, whatever the LDL comes, it is going to bind to this receptor and it will be going inside. Once it is coming inside, it is going to remove that outside protein. So here you can see this is clathrin. Clathrin is the protein, which is endocytosis. You know endocytosis? What is endocytosis, exocytosis? If something is out, it will catch whatever it needs and go inside. That is endocytosis. Exocytosis means some proteins are secreted inside the cell and finally it will give it to outside. Like insulin. Insulin will be secreted inside a cell, beta cell. The function is outside the cell. So it will give it outside. That is exocytosis. Here, this LDL is getting inside the liver. So this is called as endocytosis. So once it is coming in, something has to close it. So that closing protein is clathrin protein. Once it is coming inside, it is coated by clathrin protein. So once it is inside, this will be free. Again, it will go for the next function. Whatever has been coming inside will be degraded by lysosomes. This LDL, function of LDL, finally whatever is remnant has to be degraded in liver. Yes or no? So what is that organelle which do that degradation function? Lysosomes. In all our cells, the function of lysosome is degradation. Anything in excess, waste has to degraded only in lysosomes. So it will get degraded in lysosomes and whatever the receptor which comes along with that LDL will go back for the next function. This is called LDL receptor mediated endocytosis. So when I'm telling about degradation, there are two types of degradation in our body. Anything excess waste can be degraded only in this two ways. One is lysosomes and the another is proteosomes. You know glycogen storage disorder, you know lipid storage disorder. So all these are also can be due to defect in lysosomal action. One glycogen storage disorder, I said it is due to one enzyme in lysosome is defective. What is that? In glycogen storage disorder, I said for acid maltase, which is an enzyme present in lysosome, is not there. When it is not there, the enzyme is getting accumulated in lysosomes of heart, causing cardiomyopathy, which is seen in Pompe's disease. Yes. So any kind of anything excess and waste has to get degraded in its respective lysosomes. So here I have mentioned two types of degradation in our body. One is lysosomal degradation and one is proteasomal degradation. How to remember this two is, lysosomal degradation means dying out in old age. In old age, after age, dying. That is like lysosome. This is a normal function. Once it is going for living for a long time, after some lifespan, it has to get degraded. RBC, after 120 days, it will lyse. So all this kind of degradation will happen in lysosomes. It does not require energy. Whereas proteasomal degradation means dying in shorter time due to some conditions like suicidal. Those conditions is proteasomal degradation. For example, if protein is synthesized in our body, that protein is not properly synthesized. If synthesized properly, it will do its function. Once function has been done, after some time it will die. That is a normal process that happens in lysosome. If synthesis itself is problem, some abnormality in synthesis that will have no use in our body, that has to get degraded. That will happen in proteosomes. That is called proteasomal degradation. For this type of degradation, we need energy. Whatever has been synthesized in abnormal levels, volume, or in abnormal structure, which is having no use in our body, has to go for proteasomal degradation. So this proteasome is like a machinery which is having ubiquitin. You should know this term, ubiquitin. What is this ubiquitin means? For example, if you take some protein, this protein has to degrade because it has some amino acid defect. So for this protein to get degraded, this ubiquitin will come and bind to this protein. Ubiquitin will come and bind to this protein, and once it enters proteasome, it will be cut into small pieces for other functions. So this protein, once it is cut down, it will become each and every amino acids. Then it will go for the other functions. So this is the only two pathways present in our body for anything to get degraded. One is lysosomal degradation and one is proteasomal degradation. This is the normal routine one, and this is for most commonly for misfolded proteins. If this misfolded proteins is not going to degradation, it will get deposit in our body and causes Crayon's disease. Have you heard of this Crayon's disease? This is because this degradation pathway is not happening in our body. If anything abnormally produced, it has to be clear. If that is not clear, it will get deposited. That is called Crayon's disease. Crayon's disease is due to accumulation of misfolded proteins due to defect in proteasomal degradation. Yes, written. Okay. So this is something important in technical wise. Okay. Here I have given, since it is we are taking lipoproteins, I have given with an example of lipoprotein. So what is happening here? Can you see something as origin? From this origin, something has moved. That's why it is mobility. Something has migrated and reached this position. So this is anode, then it means this is cathode. So we have added our lipoproteins in this origin, and once we applied current from cathode to anode direction, this lipoprotein has migrated. Yes, this lipoprotein has migrated. So now you can see chylomicron at the site of origin. Next is LDL. Next is VLDL. And finally, which is towards the other end is HDL. So what is the charge towards that side? Positive charge, anode. And what is the content of HDL? You know, HDL is made up of proteins. So proteins are negatively charged. So when it is high protein, more negative charge, it is having more attraction towards positive electrode. That's why comparing to other three, HDL has moved to other side. And which is the next? VLDL, and then it is LDL, and then chylomicron. So why it is like this? VLDL, LDL, and chylomicron, which is having more of lipid content, positive charges, more in chylomicron, that is present at the top, and which is having more negative charges at the bottom towards positive current. Next is LDL and VLDL. This migration depends on the amount of cholesterol, triacylglycerol, protein, and its density. So depending on that, your chylomicron is having more positive charge, will stay towards the negative electrode, which is having more negative charge because of proteins, will stay at the other side, and your LDL is next, and then your VLDL. So this is how the migration will be. This is not important in this lipoprotein, but this technique is important. What is this technique called? What is this technique called? Like, we have added two electrode, negative electrode and positive electrode, and we have applied any substance, anything has been applied to the point. For example, serum. If you are giving serum, your protein will get separated. In protein, you have albumin, globulins, alpha 1, alpha 2, beta, gamma globulins, all will get separated by movement. So what is this technique called? This technique is called electrophoresis. What it means is separation of charged particles under the application of electric current. Separation of charged particles under the application of electric current. We can also separate not only depending on charge, we can separate depending on the mass. This is moving in horizontal way. If we allow it to move in vertical way, now it will get separate depending on its mass. Whichever is having more weight will move first. Whichever is having very less weight will move very slow. Depending on its weight, it will migrate if this electrophoresis is happening in vertical direction. If this is going to happen in horizontal direction, it will move only depending on its charge. It is called horizontal electrophoresis and vertical electrophoresis. Very important technique which we are using for patient care. So in this, in a clinical side, we will separate proteins. We will separate total protein, albumin, alpha 1, alpha 2, beta, gamma, and we will separate CSF proteins, and we will do it for CKMB. You know, CKMB is having CKMM, MB, DB. Different, everything is having different things. So that can be separated. Similarly, lipoproteins can be separated. And also we can separate hemoglobin, HbA1, HbA2, HbF, HbS, sickle cell anemia. All different isoforms of a single thing can be separated depending on this technique. They can ask you even in pathology, maybe like a pathology question also, they will give you HbA here, A2 here, S here, and then HbC here. These are the different isoforms of hemoglobin. What is this technique used for this separation? It is electrophoresis, either for proteins or hemoglobin, for creatine kinase, or for lipids, whatever it is. If we are going to separate depending on either mass or based on charge, it is electrophoresis. Okay. Okay, so next, this is important in your MCQ based one, LCAT deficiency. LCAT means lecithin cholesterol acyl transferase. Lecithin cholesterol acyl transferase. Lecithin cholesterol acyl transferase. What is the function of lecithin cholesterol acyl transferase? It will convert the cholesterol into cholesterol ester, which is present in our HDL. The function of lecithin cholesterol acyl transferase is conversion of cholesterol into cholesterol ester. That is the form where your HDL carry the cholesterol to liver. It will not carry direct cholesterol. It will carry it as cholesterol ester. For this function, we need an enzyme LCAT. So in lipoprotein, I have said you only one enzyme. Remember this, LCAT, which is in HDL for converting cholesterol to cholesterol ester. So this is important. They can ask you enzyme directly, or they can ask you what is its deficiency condition. If LCAT is completely absent, if it is completely absent, it is called as Norham's disease. Norham's disease. If LCAT is partially absent, it is called as fish-eye disease. Clear? Norham's disease, fish-eye disease, both are due to deficiency of LCAT. Hence, your HDL cannot carry cholesterol ester. So, they can ask you any of these three, whether the enzyme itself or this two deficiency names. Norham's and fish-eye. Okay, so next, this is very important topic, not only for your exam, for any kind of exam, for PG exam, MD, medicine, everywhere, this is very important. Fredrickson classification. Fredrickson classification, what is this is for? Now we are going to read about hyperlipoproteinemias. Clinical conditions where we are having more of lipoproteins. Hyperlipoproteinemia. It can be classified as type 1, classified as type 2, A and B, and type 3. The most important classification is only 3. 1, 2 and 3. Okay. So what is this first one called? What is the name? What is this? So what is increased? What is increased is hyperchylomicron. So in type 1, hyperchylomicron. It means chylomicron is higher. Hyperchylomicron. Increased chylomicrons. So increased chylomicrons means what lipid is more? Cholesterol is more or triacylglycerol is more? Triacylglycerol is more. So there will be increased triacylglycerol. Your cholesterol can be normal or mildly increased. That is no problem. But here it is increased triacylglycerol. That's why it is type 1, which is hyperchylomicronemia. From the name, you know what is the lipoprotein elevated. If you know the lipoprotein, you know what is that content. Chylomicron is having high content of TAG. Yes? Okay. So next, what is 2A? Familial cholesterolemia. This is 2A. So what is mean by hypercholesterolemia? Then what is elevated here? Cholesterol is carried in LDL. Yes, VLDL is also carrying TAG only. So Hypercholesterolemia means LDL is more. Yes. So it is hypercholesterolemia. Cholesterol will be more in LDL. So LDL is higher in this condition. So what is the lipid? Elevated cholesterol. Okay. So what is this 2? 2B. I have mentioned 2B is combined hyperlipoproteinemia. It means combination of this 1 and 2. Combined means both chylomicron and also LDL. Both are elevated. So here it is combined. Combination of 1 and 2A. What it means? There will be increased chylomicron also and also increased LDL also. So what lipid is elevated? It is both triacylglycerol is also elevated, cholesterol is also elevated. Combined means both. Okay. Clear? Clear with combined? Clear with 1, 2A, 2B? So what is 1? What is 1? Chylomicrons. So what lipid is more? Triacylglycerol is more. What is 2A? So what is elevated? Which lipoprotein is elevated? So what lipid is elevated? So what is type 2B? Combination of 1 and 2. So which chylomicron is, which lipoprotein is elevated? Chylomicron and LDL. Which lipid is elevated? Triacylglycerol and cholesterol. Okay. Clear with this? With the name you can tell. Then what is type 3? Okay, before going to type 3, what is the defect? Type 1, chylomicron is high. What is mean by chylomicron is higher? What can be the defect? Conversion of chylomicron to LDL is not happening. Clear? Chylomicron is higher means what? It has to give the triacylglycerol out and it has to convert into IDL and LDL. So now the defect is in taking the TAG from chylomicron to out. Yes. So who is responsible for taking this TAG from chylomicron to peripheral tissue? Lipoprotein lipase, which has to be activated by which apoprotein? C2. So if C2 is not functioning, if C2 is not functioning, or if lipoprotein lipase is not functioning, okay, if these two are not functioning, this TAG cannot come out. Yes or no? So it will lead to accumulation of chylomicrons. So what is the defect in hyperchylomicronemia? What is the defect in familial hyperchylomicronemia? Either defect is in C2 apoprotein or lipoprotein lipase enzyme. So if these two are not functioning, the chylomicron carrying TAG cannot come out and it will be converted to ideal. It cannot do that, hence it is responsible for increased TAG in blood. So second one is familial hypercholesterolemia. So cholesterol means LDL is high. I said after VLDL, IDL will form, IDL will convert to LDL. Now this LDL has to, go back to the liver. Yes or no? This LDL has to go back to the liver. So what can be the defect in familial hypercholesterolemia? What is the apoprotein specific for LDL, B100. C2 and E is common for all. So if B100 is defective, whatever from the VLDL it has converted to IDL and LDL, it will circulate in blood. It cannot perform the function of LDL because of absence of apoprotein. So here the defect is ApoB100 defect. You can ask then why not ApoB100 in hyperchylomicronemia? If the chylomicron ApoB100 is defect, it cannot be called as chylomicron. Only with ApoB100, whatever has been synthesized in the liver will take DAG from liver to blood. Now it has reached blood, but it cannot convert to LDL because of C21 lipoprotein lipase. But now whatever it is released, it is called as LDL. VLDL once released DAG, it is called IDL or LDL. Now this has to give that cholesterol to peripheral tissues, which is recognized by the peripheral tissues will recognize this is LDL by ApoB100. If that is not going to recognize ApoB100, this will circulate in blood. So that is the reason for ApoB100 defect in type 2A, that is hypercholesterolemia. In type 2B, it is combined. It can be any of this. It can be any of these three or both. ApoB100, C2 defect, any defects. So till this 3, you have the logic of name and also the defect. So what about this type 3? This beta lipoproteinemia. From the name you cannot recognize what is this. This beta lipoproteinemia is due to defect in ApoE receptor defect. Where is ApoE receptor present? Liver. In your liver, you will have the receptor. If this receptor is not there in liver, whatever in the blood, your LDL or IDL remnant cannot go back to liver for degradation. So there will be circulation of, remnant VLDL, remnant IDL and also remnant VLDL in circulation. This is due to ApoE receptor defect. Once it has done its work, it has to go back for degradation that it cannot perform. So the remnant part is circulating in the blood. So here you can see both increased cholesterol and also triglycerol. But the significant is in this hyperchylomicronemia, our blood triglycerol can go to more than 500 milligram per deciliter. The normal triglyceride level in blood is 150. Less than 150 milligram per deciliter is normal level for TAG in our blood. But in this type 1 condition, it will go to up to 500 milligram per deciliter. But in this condition and all, it will not be elevated that much, because it is only remnant. Here not elevated that much, but more than normal it will be there. Here in some patient I have seen 1500 milligram per deciliter of triglyceride. When I take and centrifuge that plasma, the plasma will looks like this. Once I have collected the sample from the patient in red tube, yes, I want serum for cholesterol estimation. Once I collect the patient sample in red tube, after centrifuging, it will looks like this. This is after centrifuging, this is cells. Above my serum should be clear. Actually, it should be clear, but this will be full of thick, white, cloudy substance. This is nothing but triacylglycerol. It will form a milky white, thick, viscous substance at the top of the cells. It will not allow the serum to separate. So in this condition, on visible things, we can tell this can be more than 500, more than 1000. So this kind of that we will see in hyperchylomicronemia patients. Okay, all these 4 type 1, 2A, 2B, 3, totally 4. All these are hyperlipoproteinemia condition. In hypolipoproteinemia condition, I have said tangentious disease. HDL is less. Okay, this 5 condition is very, very important. They can ask you any questions. Okay, so this I have already said, tangential disease, which is due to defect in ABCA1 gene defect. So what is this? Can you see some nodules? Yes. Any idea what is this? They are lipid deposition. They are fat deposition in various sites. This is tendon xanthoma. This is tendon xanthoma. This is follicular xanthomas. From the hair follicles, each and every hair follicle tip will have that fat deposition. This is Palmer. In all and every creases you can see this is Palmer xanthoma. This is tubulo-eruptive xanthomas. In all joint positions you can see this is tubulo-eruptive xanthoma. So if you see any this kind of a picture or a clinical case with mentioning as xanthoma, it is hyperlipoproteinemias. Okay, only this is caused by fat deposition. So what fat has been deposited? So this is very important. Tendon xanthoma, cholesterol will be deposited. Okay, in eruptive xanthomas, triglycerol will be deposited. In Palmer and tubulo-eruptive xanthoma, chylomicron remnant. It means chylomicron remnant will have cholesterol. Cholesterol increased. This milky plasma only I have said. When triglyceride is going more than 500, I will not get serum. I will get thick white viscous plasma. Chylomicron is increased. It means triacylglycerol is increased. Patient can also present with acute pain abdomen in increased TAG. Tendon means your cholesterol. It means LDL can move out and it can deposit the cholesterol in peripheral tissue. That peripheral tissue is tendon. Whereas in eruptive Palmer conditions, your chylomicrons will give that cholesterol to peripheral tissues. All this can be seen in very, very high level of cholesterol. Even if we have some nodule, either in any of the joint creases, anywhere, it means some of our lipid profile is abnormal. Tendon xanthoma, Palmer. Why I have given with the picture is, if I tell the term, it is easy, but in exam, if they ask you with picture, you don't know what is this. So if they are giving this and asking you what is the condition, then it means it is Palmer xanthoma, triacylglycerol is increased. Okay. Okay. So we'll take some 10 minutes break. Come back by 10:30 and we'll continue few more points and we'll go for fat metabolisms. So any doubts till this? Any doubts? Anything? Am I going very fast? It's fine. So lipoprotein lipase. What is the function of lipoprotein lipase? We were discussing. What is the function of lipoprotein lipase? Yes. So it is breaking up actually the triacylglycerol. It is breaking triacylglycerol into glycerol and 3 fatty acid, which will go to adipose tissue again for storage. So this is the chylomicron. if it is from intestine, or it is VLDL, which is having ApoC2. So this ApoC2 is recognized by lipoprotein lipase, which is present in our blood endothelial cell. Okay. So this is the lipoprotein lipase recognizing the ApoC2 present in VLDL or IDL, whatever, chylomicrons. So this is lipoprotein lipase. Then what is this structure? Yesterday, when I'm discussing about mucopolysaccharides, I said something which is anchoring lipoprotein lipase present in the blood endothelial cell, which is anchoring lipoprotein lipase. Find out. Chondroitin sulfate or dermatone sulfate or heparin sulfate or heparin or hyaluronic acid out of this glycosaminoglycan, which is, here you can see polysaccharide chain they have mentioned, but you know what is that polysaccharide? What is that polysaccharide? What is that glycosaminoglycan, which is anchoring lipoprotein lipase? This is heparin sulfate. Heparin sulfate is supporting the lipoprotein lipase for the function. Clear? So this is heparin sulfate. So now what is the role of heparin? What is the role of heparin? If we inject heparin, I have given heparin to this patient. So what this heparin will do? This heparin, now it will cut the lipoprotein lipase from my heparin sulfate. So if it cuts this anchoring part and this lipoprotein lipase will circulate now in free, in blood. Yes, because from the endothelial cell, heparin sulfate is supporting lipoprotein lipase. I have added heparin injection, which has cut this lipoprotein lipase. Now this will be circulating free in the blood. So now if you take blood sample of this patient and if you estimate lipoprotein lipase, this will be elevated. Then it means our heparin is working. Clear? If I inject a heparin to a patient, I have to find out whether my heparin is functioning or not. So for that, what I should do, I will take the blood sample of that patient and I will estimate lipoprotein lipase. If this has cut lipoprotein lipase, obviously it will cut other cholesterols, fat, whatever has been accumulated in that blood vessel also. So the function of heparin is preventing that clotting. Yes or no? So it will not allow anything to get clotted. Everything will be circulating independently in the blood. So this is the one way of estimating my function of heparin. But this is important. The lipoprotein lipase is anchored by heparin sulfate. Okay. So here you can expect 3 questions. One is heparin, one is heparin sulfate, and the another is ApoC2 and lipoprotein lipase. So you know what is lipoprotein lipase. Do you know what is hormone sensitive lipase? So this is blood. In blood, you know, you have lipoprotein lipase. The function of lipoprotein lipase is removing of cleaving of TAG into glycerol and fatty acid. Now this will go back to adipose tissue where these two can combine again to form triacyl glycerol. Clear? Okay, so you know lipoprotein lipase is present in blood endothelium. Correct? Okay, so what is its function? Cleaving of TAG into glycerol and fatty acid in blood. This you know, right? Okay. So this enzyme will be active in fed state or fasting state? When this function will happen? Only after taking food, our body will be having more of fat. Only then it is going to blood for storage. So this is enzyme which is activated by the hormone insulin. Clear why it is insulin? All this lipoprotein activity of storing the fat to the peripheral tissues will happen only when we have high diet, high fat diet. So this is activated by insulin. This enzyme is activated by insulin. So these points you know about lipoprotein lipase. Then what about hormone sensitive lipase? Hormone sensitive lipase itself the same activity, cleaving of triacylglycerol. So this is present in adipose tissues. The function of hormone sensitive lipase is same, cleaving of triacylglycerol into glycerol and fatty acid, but the location is different. This enzyme is present in adipose tissue. So in adipose tissue, whatever the hormone sensitive lipase is present will cleave the stored triacylglycerol into glycerol and fatty acid. Okay. Same thing, it is also cleaving TAG into glycerol and fatty acid in adipose tissues. So when this will happen, when will our stored fat will get cleaved? During fasting condition. So what is the hormone which is activated? Activating hormone sensitive lipase is glucagon. So this happens in fed state. This activity happens in fasting state. So this enzyme will be active in dephosphorylated state, whereas hormone sensitive lipase will be active in phosphorylated state. Understood? What is lipoprotein lipase, hormone sensitive lipase? Apart from that, we have hepatic lipase, intestinal lipase. Everything is cleaving of triacylglycerol in its respective organs. When it comes to metabolism and regulation of blood glucose, these two are very important, lipoprotein lipase and hormone sensitive lipase. Okay, function wise same, which is present in adipose tissue, hormone sensitive, which is present in blood vessel, lipoprotein lipase. So which is activated by the hormone glucagon, which is going to happen in fasting condition. Only during fasting, whatever has been stored will get broken down to give blood glucose. So it is hormone sensitive lipase. Clear? Yes. So shall we go to metabolism? Okay. So fat metabolism. During fed state, during fasting state. So which is going to happen during fed state is fatty acid synthesis. Fatty acid synthesis will happen during fed state. What is going to happen during fasting state is breakdown of fatty acid, that is called as beta oxidation. Oxidation of fatty acid means cleavage of fatty acid. So for fasting, it is beta oxidation of fatty acid. Fed state, fasting state. Fed state la for fatty acid synthesis will happen. Fasting state la beta oxidation of fatty acids will happen. So what enzyme will be active in fed state? Insulin. So insulin activates this pathway. Glucagon, cortisol, epinephrine, norepinephrine will activate beta oxidation of fatty acid. So your fatty acid synthesis, the rate limiting enzyme, the one rate limiting enzyme for fatty acid synthesis is acetyl-CoA carboxylase. This is the rate limiting enzyme, acetyl-CoA carboxylase, which is active in which state? It is active in dephosphorylated state because it is happening by action of insulin. Okay. So what is the rate limiting enzyme for beta oxidation of fatty acid? It is carnitine palmitoyl transferase 1. We have 2 also, but the rate limiting enzyme which is involved here is 1. Shortly CPT1. So this enzyme is active in phosphorylated state. Yes. Okay. So now you know, in fasting condition, there are many pathways which will help us to maintain blood glucose from the glucose metabolism also. We have read about glycogenolysis, which is happening in fasting state, gluconeogenesis, which is happening in fasting state, and in fat you read. Now we are going to see beta oxidation of fatty acid, which is going to happen in our fasting state, and also ketone body synthesis will happen during fasting state. So all these four will happen during fasting state. So now we should know which will come first, which will come next. What is well-fed state? 2 to 4 hours is fed state. So after 4 hours, we call them as fasting. So the first 4 to 16 hours, your glycogenolysis will maintain your blood glucose. Okay, 4 to 16. Next 16 to 48 hours, nearly for 2 days, your gluconeogenesis will give glucose for energy and maintaining blood glucose. So after 48 hours to 72 hours, only beta oxidation will happen. So it means whatever we are going to have food stored as fatty acid will get cleaved after 3 days of fasting. Till that, whatever has been stored fat will be stored. Clear? Whatever excess calories we are going to take it as fat is going to get stored. So that stored fat will be lysed. Fatty acids will be lysed only after 48 to 72 hours of fasting. So if we are not going to take any food even after 3 days, we can survive for few more days with help of ketone bodies. So it is ketone body synthesis. So this is called yearly fasting. 4 to 16 hours is called yearly fasting, and this is called as fasting 16 to 48 hours. When we are going to fast for more than 16 hours, the actual fasting happens, fasting. When it is more than 48 hours, it is called as starvation. This is late starvation. Okay, so this is how one by one the reaction will happen. It's not like after 4-5 hours anything will come and give the blood glucose. Still first 4 to 16 hours glycogenolysis only will happen. Once that glycogen has been depleted, now we will gluconeogenesis, conversion of pyruvate to oxaloacetate to reverse of glycolysis will come. Next comes beta oxidation of fatty acid, and then comes the final thing is ketone body synthesis. Okay, so you know this two already we have discussed. Now we'll discuss fatty acid synthesis and then beta oxidation. Okay, first we will go for fatty acid synthesis. Okay, fatty acid synthesis. Acetyl-CoA. Have you heard this? Acetyl-CoA yesterday? In which pathway? What is the acetyl-CoA is a substrate for which pathway? TCA cycle. Where it is happening? Mitochondria. Your fatty acid synthesis is going to happen in cytoplasm because this is decided by the location of that enzyme. That will decide in which organelle the pathway has to happen. So fatty acid synthesis enzyme, the rate limiting enzyme acetyl-CoA carboxylase will be present in cytoplasm. But you know acetyl-CoA is in mitochondria now. Yes. So that acetyl-CoA which is present in mitochondria has to enter cytoplasm for fatty acid synthesis. So acetyl-CoA combines with oxaloacetate to form citrate. Is this the step in TCA cycle? In TCA cycle, have you seen oxaloacetate combines with acetyl-CoA to form citrate and then isocitrate, everything. So now this citrate can cross the mitochondrial membrane. So this citrate, whichever has been formed in the mitochondria in TCA cycle, will come to cytoplasm. Once it come back to cytoplasm, this is again cleaved back to oxaloacetate and acetyl-CoA, and this acetyl-CoA is used for fatty acid synthesis. So for that acetyl-CoA to carry from mitochondria to cytoplasm, we need citrate. Acetyl-CoA cannot cross the membrane. Citrate can cross the membrane, so it convert to citrate. Once it come back to cytoplasm, again it convert back to oxaloacetate and acetyl-CoA, and fatty acid synthesis happens. Okay, so what is this called? This kind of shifting one from one side to other side. Yesterday we saw some two things for transferring NADH from cytoplasm to mitochondria. Malate, aspartate, shuttle, glycerolidate 3-phosphate, shuttles. Yes, that is shuttle means shifting of one compound from outside cytoplasm to mitochondria, mitochondria to cytoplasm. So here that role is by citrate. So here citrate, this is an important point. Citrate act as a transporter of acetyl-CoA. Citrate, remember this because you will get one more transporter in beta oxidation also. Why? Whatever has been fatty acid is happening in cytoplasm, beta oxidation will happen in mitochondria. So whatever has been produced here has to go back. So again you will read one more transporter. This is citrate transporter in fatty acid synthesis. Okay. First thing you should know where it is happening. Second thing you should know what is the transporter. And third, this is very important because conversion of acetyl-CoA to malonyl-CoA. This is the first step in fatty acid synthesis. Not all steps are needed. Not at all needed. Only this step is essential. This is the first step, conversion of acetyl-CoA to malonyl-CoA by the enzyme. What is that enzyme? Rate limiting enzyme. What is the rate limiting enzyme for fatty acid synthesis? Acetyl-CoA carboxylase. Why it is called acetyl-CoA carboxylase? Acetyl-CoA and this CO2 is added. Yes. So that's why it is called acetyl-CoA carboxylase. This is actually 2 carbon. You know acetyl-CoA is 2 carbon. In malonyl-CoA, it is becoming 3 carbon. Slowly, fatty acid synthesis function is to adding up of carbons. You know, we have short chain fatty acid, medium chain fatty acid, long chain and very long chain. Till 20 to 22 carbons, it can add up. So in first step, 2 carbon has become 3 carbon. In successive steps, it will add 4, 5, 6, 7. And depending on that, it will stop at 8 carbon or 10 carbon, 12 carbon. So we will name each and every number of carbon atom as palmitic acid, oleic acid, lactic acid, like that, different names for fatty acid. So fatty acid synthesis means we are going to add up the carbon and we are going to store it as a fat. Okay, so this is important step because this is rate limiting enzyme. So acetyl-CoA carboxylase, it will come under which classification of enzymes? Carboxylase. Check and tell me. If when you are reading metabolism, if you compare this, you will never forget. Ligase? Ligase or ligase? Ligase. Okay. So is there any coenzyme required for carboxylase? Biotin and ATP. So can you see ATP here? Can you see biotin here? So for all carboxylase, biotin and ATP are the coenzymes. Clear? So that's why this step is important. The first is rate limiting enzyme, and they can ask you what are the cofactors needed for this enzyme, or they can ask you it will come under which classification of the enzyme. Okay. So this is not much important, but this is the enzyme structure involved in fatty acid synthesis. So if I zoom, you can see synthase, transacylase, hydratase, reductase, thioesterase. So this is called multi-subunit complex. Different enzyme all coming together to perform a single function, that is fatty acid synthesis. This is the enzyme which is enzyme complex involved in fatty acid synthesis. One thing you should know is what all enzyme involved. Synthase, transacylase, hydratase, reductase, thioesterase. Just remember this 5 complex involved in fatty acid synthesis. So all this function is one by one adding the carbon to that base acetyl-CoA. 2 will become 3 carbon, 4 carbon, 5 carbon, till that it can add 16, 17 carbons. Okay. So this is the enzyme fatty acid multi-enzyme complex. So in this complex, we are having synthase. We are having hydratase. We are having reductase. We are having transacylase. Yes, transacylase, and we have thioesterase. If you don't remember this 5 also, no problem, but it is a multi-enzyme complex. It is not a single enzyme. Remember that much. So regulation of fatty acid, acetyl-CoA carboxylase, you already know. In regulation means hormonal regulation and allosteric regulation. Whatever may be the compound, you have to talk about 2 regulation, allosteric regulation or hormonal regulation. You know, in hormonal regulation, insulin activates acetyl-CoA carboxylase and keeps this enzyme in dephosphorylated state. And glucagon inhibit this enzyme. Glucagon, cortisol, epinephrine, norepinephrine, catecholamines. So this is hormonal regulation. Then what about allosteric regulation? What about allosteric regulation? You know acetyl-CoA, which is 2 carbon, with help of the very important enzyme acetyl-CoA carboxylase, it will become malonyl CoA, which is 3 carbon. After with subunit, multi-subunit enzyme complex, that hydratase, reductase, with all this, it will form some 10 to 15 carbon long chain fatty acid. Okay, so this is that fatty acid synthesis. If this fatty acid is more, if it is very high, what this end product will do? This will inhibit the rate limiting enzyme. This is called as allosteric inhibition or feedback inhibition. The product is more, so it will inhibit the enzyme which is responsible for the synthesis. Hormonal regulation, and who is inhibiting acetyl-CoA carboxylase means long chain fatty acid. Long chain fatty acid which has been synthesized from this pathway will inhibit the rate limiting enzyme. The same thing, it will be inhibited by long chain fatty acid also. So who can activate this? Always the substrate, that is acetyl-CoA. Here, acetyl-CoA. Always the substrates are activator for the enzyme and the products are inhibitor for the enzyme. Any pathway, if it is substrate forming product with help of an enzyme, the substrate will activate. I am here, start the reaction. But this product will inhibit. The product will tell I am already very high, don't want any more reaction. So this will inhibit this enzyme. This is called allosteric inhibition. Yes, done. Okay. Next, metabolism of odd chain fatty acids. All even chain fatty acids will go to beta oxidation of fatty acids. Odd chain fatty acid, this is the metabolism. So what is the significance here? They can ask minimum 3-4 MCQs from this single pathway. So it is important. Okay. So yesterday, in one pathway in carbohydrate metabolism, I said propionyl-CoA can be a substrate. There is one pathway which has multiple substrates. I said all carbohydrates, glucose can convert to fat, but fat cannot convert to glucose except one, because that one fat is involved in gluconeogenesis, that is propionyl-CoA. So what are the substrates for gluconeogenesis? Check and tell me. What are the substrates for gluconeogenesis? Have you written this fatty acid in that as a substrate? All glucose can convert to fat. You know, as triacyl glycerol, whatever may be the glucose, glyceraldehyde, glycerol, it will go for triacyl glycerol, fat. But all fats cannot be converted to glucose except propionyl-CoA, because propionyl-CoA can be a substrate for gluconeogenesis. Okay. So how it is intermediate for gluconeogenesis? For lactate, you know, Cori cycle is happening. For alanine, you will read in protein metabolism, one more cycle is happening. Propionyl-CoA is also a substrate. How it is going to enter to gluconeogenesis? We'll see here. So for odd chain fatty acid, 1, 2, 3. 3 carbon. That's why it is odd chain. So propionyl-CoA, first is propionyl converted to propionyl-CoA. CoA means acyl-CoA with help of enzyme acyl-CoA synthetase. Propionyl-CoA will form D-methylmalonyl-CoA and then L-methylmalonyl-CoA and finally succinyl-CoA. So forget this two, no need. Remember, propionyl-CoA, after undergoing metabolism, it forms succinyl-CoA. Clear? This succinyl-CoA, have you read succinyl-CoA succinate in any of the reaction yesterday? TCA cycle? Yes. Alpha ketoglutarate convert to succinyl-CoA, succinate, and then fumarate, malate, oxaloacetate. This oxaloacetate is involved in gluconeogenesis. Yes or no? Pyruvate will convert to oxaloacetate. Yes or no? Pyruvate will convert to oxaloacetate. This oxaloacetate is going to be converted to phosphoenoyl pyruvate in gluconeogenesis. Go to gluconeogenesis page. Open that. Taken. Can you see pyruvate is converting to oxaloacetate? Yes. Oxaloacetate is converted to phosphoenol pyruvate. Yes. Can you see that? Now this oxaloacetate is converted to phosphoenol pyruvate and go to gluconeogenesis for synthesizing of glucose. Yes. So this oxaloacetate can be formed from two What are they? One is from pyruvate and one is from TCA cycle intermediates. Yes or no? Because oxaloacetate is involved in TCA cycle also. Alpha ketoglutarate, succinyl-CoA, succinate will form fumarate and fumarate will form malate. Malate can also form oxaloacetate. Pyruvate can also form oxaloacetate. Malate can also form oxaloacetate. Yes or no? So for synthesis of malate, you need fumarate. For synthesis of fumarate, you need succinate. For synthesis of succinate, you need succinyl-CoA. For synthesis of succinyl-CoA, you need propionyl-CoA. That's why propionyl-CoA is one of the substrate for gluconeogenesis. Understood? Why fatty acid, short chain fatty acid and TCA cycle and gluconeogenesis, everything is linked. Propionyl-CoA will form succinyl-CoA. By this metabolism, this succinyl-CoA is forming fumarate malate of TCA cycle that form oxaloacetate. Only if oxaloacetate is there, gluconeogenesis can happen. Clear? Okay. So this is one important question. Propionyl-CoA to succinyl-CoA, where all it is related. TCA cycle it will go, gluconeogenesis it go. So anything else is important? Okay. Carboxylase. Carboxylase means what is the cofactor? Biotin and ATP. They can ask you propionyl-CoA carboxylase, what is the cofactor? Okay. And then isomerase racemase. What it means? What is racemase? It will come under which classification? Racemase will come under which classification of enzymes? Epimerase, racemase, or also isomerase. Yes, only structurally these two are different. Both are methyl malonyl-CoA only. Only it is in D form, it is in L form. So it is called either racemase or isomerase. So what is the coenzyme involved in methyl malonyl-CoA isomerase? Actually, this is isomerase or mutase. In many books, it will be mentioned as methyl malonyl-CoA mutase. So what is the coenzyme involved in methyl malonyl-CoA mutases? B12. So if we are deficient of B12, succinyl-CoA will not be formed. If succinyl-CoA is not formed, you know, TCA cycle will not happen, gluconeogenesis will not happen. That's why this is important. They can ask you B12 deficiency can cause decreased blood glucose level during fasting. B12 deficiency can cause decreased blood glucose level during fasting. During fasting, gluconeogenesis has to happen. For gluconeogenesis to happen, you need oxaloacetate. For oxaloacetate, you need succinyl-CoA. To form succinyl-CoA, we need B12 as a coenzyme. So this is important question. This B12, methyl malonyl-CoA mutase. The cofactor involved here is B12. They can ask you what is the other vitamin involved in this. It is biotin in propionyl-CoA carboxylase. So these two vitamins are important in short chain fatty acids. And always you should remember what is the substrate, propionyl-CoA. What is the end product, succinyl-CoA. Okay. Yes. Okay. Just 1 minute. Have a look about fatty acid synthesis or chain fatty acids, lipoproteins, everything. Then we'll start beta oxidation. If we switch off this light, will it be more clear? Where is the switch? Can you just try, anyone? Just try with one light. If we switch off, whether this is more clear. Oh, but this is it better for you? இல்ல ஆன் பண்ணிருங்க. அவங்களுக்குமே லைட் இல்ல இல்ல, அவங்க எழுதுறதுக்கு லைட் இல்ல இல்ல, அதான் அதனால ஆன் பண்ணிடலாம். நான் இது ஆஃப் பண்ணா ஃபர்ஸ்ட் 2 rows அஃபெக்ட் ஆகும். ஆன் பண்ணி so yes, next. So what is this? We are going to see about beta oxidation of fatty acids. Beta oxidation. What is mean by beta oxidation of fatty acid? Breakdown of fatty acids. So when it will happen? Fasting state. Tell me, what is the rate limiting enzyme? What is the rate limiting enzyme? Carnitine, palmitoyl transferase 1, CPT1. So this is the first step where fatty acid synthesis has happened. In which organelle? It happened in which organelle? Cytoplasm. Fatty acid synthesis happened in cytoplasm. Beta oxidation of fatty acid will happen in mitochondria. So the long chain fatty acid after fatty acid synthesis has been formed in the cytoplasm. Yes, your fatty acid has been formed in the cytoplasm. Now this has to enter inside mitochondrial matrix. So this is the outer mitochondrial membrane and this is the inner mitochondrial membrane. Now this free fatty acid has to enter here. It cannot enter as it is. You know, acetyl-CoA cannot enter. It needs some transporter. So this free fatty acid, okay. Before that, I forgot to tell one point here. In fatty acid synthesis, it is not an energy producing pathway. It will utilize the energy. In which pathway it has utilized the energy? In first step. Yes. So in this, it has utilized 1 ATP. That is the total ATP that is utilized in fatty acid synthesis. Only 1 ATP has been utilized in fatty acid synthesis. No ATP or NADH generated in fatty acid synthesis. They can ask you whether it is energy producing pathway or utilizing pathway. It is utilizing pathway, utilizes only 1. So for beta oxidation, in the first step, ATP has converted to AMP. Triphosphate has converted to monophosphate. So it means how many phosphate has been utilized? 2. So it is utilizing 2 ATPs, right? Even though one is converted to AMP, how much utilizes? 2. So it is utilizing 2 ATPs in the first step. This is the only step where it utilizes. No energy will be produced at the end of beta oxidation. No, it produces energy, but utilization is only 2. So in first step, free fatty acid has been converted to acetyl-CoA. Now this acetyl-CoA has entered to intermembrane space. Intermembrane space. This is intermembrane space. After this, it cannot cross the inner mitochondrial membrane. So this acetyl-CoA combines with carnitine. So for this acetyl-CoA to enter, it will enter as it is. But once it is entered, this has to combine with carnitine to form acyl-carnitine. So this conversion needs an enzyme, that is the rate limiting enzyme. of beta oxidation, which is carnitine palmitoyl transferase 1. So what is the function of carnitine palmitoyl transferase 1 is combining carnitine with acyl-CoA to form acyl-carnitine. Where it is present, they can ask you intermembrane space, outer mitochondrial membrane, inner mitochondrial membrane, and matrix. You should not confuse. It is present in outer mitochondrial membrane because your CPT2 is present in inner mitochondrial membrane. It has no function in beta oxidation. So the rate limiting enzyme CPT1 present in outer mitochondrial membrane. The function is formation of acyl-carnitine. Now here your carnitine is the transporter for acyl-CoA. What is the transporter in fatty acid synthesis? What is the transporter in fatty acid synthesis? Citrate is the transporter. In beta oxidation, carnitine is your transporter. So here acyl-carnitine will cross inner mitochondrial membrane with help of an enzyme carnitine acyl-carnitine translocase. Translocase. So this is coming under classification 7. What I said, translocase means something is going in, something is coming out. So here in this translocase, you can see acyl-carnitine is coming in, the free carnitine is coming out. That's why it is called translocase. Okay, so with help of this enzyme, whatever the acyl-carnitine form will enter mitochondrial matrix. Yes, it has reached mitochondrial matrix. So after entering into mitochondrial matrix, you want only acyl-CoA for beta oxidation. Carnitine has to go back to its own position. So here it will get degraded into free. It get free into carnitine and acyl-CoA. This carnitine will go back here. Again, it will carry another acyl-CoA and come back and it circulates. Whatever the acyl-CoA which has entered mitochondrial matrix will now go for beta oxidation. So till this, only the acyl-CoA after that fatty acid synthesis in cytoplasm has reached mitochondria. Beta oxidation has not yet happened. Now only the transfer has been happened. So in this transfer itself, we have many MCQs. First is, how many ATP utilized? 2 ATP. So what is the rate limiting enzyme for beta oxidation is? For beta oxidation itself, it is CPT1. For transferring enzyme is CPT1, that is the rate limiting enzyme. If this is not functioning, your acyl-CoA cannot enter. If it is not entered, beta oxidation will not happen. So this is the rate limiting enzyme for beta oxidation also. Okay, so this is the rate limiting enzyme for beta oxidation also. So now this acyl-carnitine with help of carnitine acyl-carnitine translocase. Carnitine acyl-carnitine translocase, because acyl-carnitine is coming in, carnitine is moving out. So that's why it is translocase. Once it enters, your acyl-CoA will enter to beta oxidation. Now beta oxidation going to start. So this is the first step which we have already discussed. Carnitine transporter. With help of carnitine transporter, your acyl-CoA has entered mitochondria. Okay. So now you can see many steps. This is not important. Okay. You know how it has entered. Each and every step not at all important. What you should know is how many NADH, sorry, this is FADH, and how many NADH has been produced. It is happening in fasting state. Beta oxidation happens in fasting state. So it has to provide energy or it has to provide glucose. For glucose, we have gluconeogenesis, glycogenolysis. So this is happening after how many hours of fasting? Beta oxidation happens after how many hours? 48, around 48 to 72 hours. So at that time, we don't have glucose, but we need ATP, energy for brain, liver, kidney, skeletal muscle, cardiac muscle, for all muscles. So it has to provide more energy. So in one beta oxidation cycle, we will get one NADH2 and one 1 NADH and 1 FADH. Okay, at the end of beta oxidation, you know 2 ATP has been utilized. It's okay if it is utilizing 2 ATP even during fasting, it is going to give you more ATP than more than that. So it is giving 1 NADH and 1 FADH for one cycle of beta oxidation. So how many cycle will happen? If I'm telling for one cycle, it will give 1 ADH, 1 NADH and 1 FADH. It means there are going to happen multiple cycles of beta oxidation. So what is mean by beta oxidation? I said this total cycle is not important, but you should understand what is happening in beta oxidation. So this is the fatty acid. How many chains? How many carbon? It can be any types. If I'm going to take your 20 carbon fatty acid, your 20 carbon fatty acid is going to enter beta oxidation of fatty acid. Okay. Just understand, with fatty acid synthesis, we have made long chain, very long chain, medium chain fatty acids. Those fatty acids now undergoing beta oxidation to give you energy. So 20 carbon, this is only for example, I am taking 20 carbon fatty acid is now going for beta oxidation. It is going to cut. How it will going to cut is, at the end, towards the end, you will get 2 carbon acetyl-CoA plus 18 carbon fatty acid. Clear? After one cycle of beta oxidation, whatever may be your fatty acid chain, it will give you 2 carbon acetyl-CoA and remaining chain of fatty acids. This is one cycle. Clear? So now you have here 18 carbon fatty acid. Now this will go to beta oxidation and it will give you 2 carbon acetyl-CoA with 16 carbon fatty acid. Now this 16 carbon again enter to next cycle of third cycle of beta oxidation to give you 2 carbon acetyl-CoA with 14 carbon fatty acid. So like this, one by one, it is going to release acetyl-CoA at each and every step. Why it has to cleave like this? Because only TCA cycle can happen with presence of acetyl-CoA. Yes or no? Acetyl-CoA is a substrate for your TCA cycle. Your TCA cycle will give you more ATP, 10 ATPs in one cycle. So like this, it will cleave 2 carbon acetyl-CoA and each carbon will enter to TCA cycle and give you more amount of ATPs. So this is the concept of beta oxidation happening. This pathway enzyme not needed. You should know whatever may be the number of carbon, how much of energy we are going to get. So with this, is it possible to calculate how much of energy for each and every fatty acid? Yes, for this we have one small equation. If you remember that in exam you can write beta oxidation. So you know in beta oxidation cycle itself you will get 1 NADH and 1 FADH and then you will get finally acetyl-CoA that will give you 10 ATP. Yes or no? So how many beta oxidation cycle happening? So 1 NADH means 2.5, 1 FADH means 1.5, 2, 3, 4. Okay, 4 ATP from beta oxidation cycle itself, and with this acetyl-CoA will give you 10 ATP, so totally 14. Again it will go another, again it will be 24, 34. Not 24, another cycle, here you will get another ATPs, 4 ATPs. So for beta oxidation, I will give you some 2 equations, 2 small formulas. To find out number of beta oxidation cycle. To find out number of beta oxidation cycle, you have to divide number of carbon atoms in the fatty acid by 2 minus 1. This is to find out how many cycles. If it is 20 carbon, first with example I have said only 20 carbons, no? So if I take 20 carbon fatty acid divided by 2 minus 1, it is 10 minus 1, 9. So totally 9 cycles of beta oxidation will happen. And in one cycle of beta oxidation, I will get 1 NADH and 1 FADH. So I will get 4 ATP in one cycle of beta oxidation. Yes. So here for 20 carbon fatty acid, I got 9 cycles of beta oxidation. So 9 into 4, 28. So only from the beta oxidation itself, I will get 28 ATPs. First point clear? So only this is not our energy source. The major energy source is to find out how many acetyl-CoA we will get from that fatty acid. So for this, it is very simple. Number of carbon atom by 2. That's all. Number of carbon atom by 2. It will tell you how many acetyl-CoA. Number of carbon atom by 2 minus 1 will tell you how many beta oxidation cycle. So here, the same 20 carbon. 20 by 2, 10. So you know 1 acetyl-CoA will give you 10 ATP in TCA cycle. Yes, 1 acetyl-CoA will give you 10 ATP in TCA cycle. But with 20 carbon fatty acid, I got 10 acetyl-CoA. So 10 into 10, 100 ATPs I will get from acetyl-CoA. So now as a total, how many ATPs I will get from beta oxidation for 20 carbon fatty acid is 100 plus 28, 128. This is not going to get completed. In first step, we have utilized 2 ATPs. Yes or no? In first step with CPT1, we have utilized 2 ATPs. Totally, my 20 carbon fatty acid will give me 126 ATPs. I will repeat it again and I will give you, you will calculate for other fatty acids. Okay. Written? Done? Just listen again. The first step is conversion of fatty acid to acyl-CoA in cytoplasm. This has to enter mitochondria. In this first step of entering into mitochondria, 2 ATPs has been utilized. So whatever may be the acyl-CoA is entered, it will undergo series of reactions to give you 1 acetyl-CoA, and this, if this is X, if this is X carbon, you will get X minus 2 acetyl-CoA and X minus 2 fatty acyl-CoA and 1 acetyl-CoA. Minus 2 because you got 1 acetyl-CoA. So this will become minus 2. So in this one cycle, you will get 1 NADH and 1 FADH. So totally 4 ATPs. From the cycle itself. At the end, you got acetyl-CoA. The fate of acetyl-CoA is it will go to TCA cycle to give you another 10 ATP. So this is for first reaction. Now here you got X minus 2. This will again enter to that same cycle. Again, in the second step, you will get another 4 ATP and 1 acetyl-CoA. Again, it will go to third step. Again, 4 ATP with 1 acetyl-CoA. So like this, how many cycles it is going to form will be decided by this formula. If it is 20 carbon, it is 20 carbon by 2 minus 1 means 9 beta oxidation cycle will happen. 9 beta oxidation cycle, and finally you will get 9 plus 1, that is 10 acetyl-CoA. Clear? Why it is 9, why it is becoming 10? At the last you will get 4 carbon entering beta oxidation. When 4 carbon are entering beta oxidation, it will get cleaved into 1 plus 1 acetyl-CoA. That's why whatever may be the beta oxidation cycle, your number of acetyl-CoA will be 1 higher than that. So here it is number of carbon by 2 minus 1. Here is number of carbon atom by 2. So if you calculate for 1 beta oxidation 4 ATP, depending on your number of carbon atom, it is 9 into 4, 28. if number of acetyl-CoA, you got 10. One acetyl-CoA will give you 10 ATP in TCA cycle. So total energy I will get is 10 into 10, 100. So 100 plus 28, 128. But in this first step, 2 ATP has been utilized. So minus 2, finally 126. So now can you make it for 16 carbon fatty acid? If a 16 carbon fatty acid is entering into beta oxidation, how many ATPs we will get? 16 carbon fatty acid. In 16 carbon fatty acid, what is the first equation? You have to find out number of beta oxidation. It is 16 by 2 minus 1. 8 minus 2, 7 cycles. Next is you have to find out number of, you write in your paper, don't see this. You calculate and tell me. Number of acetyl. Okay, wait, others will wait for others. Done. How much? 106. Everyone got 106? Yes? Okay. So can you do it for 14 carbons? Do it for 14 carbon fatty acid. How much energy it will give you? (22) 92. What about others? We'll see. (92). Everyone got 92? Yes. So in exam, if they ask you to calculate for any fatty acid, you can calculate the amount of ATPs. Yes. Don't forget to subtract that 2. In option, they will give you 108 also, 106 also. You will be very curious towards 108. Don't forget to subtract that -2. Okay. So yes, next, ketone bodies. So when ketone body synthesis will happen, fed state or fasting state? Fasting state. Since it is synthesis, it does not mean it is going to happen in fed state. It is synthesizing ketone bodies. So this is going to happen in fasting state. So why we need ketone bodies? Okay, I will make RBC, heart, brain, liver, kidney, everything will be similar to liver. So I will make like this. What is the energy source for all this four during fed state, fasting state, and starvation state? So now we will understand why we need to read ketone bodies. Okay. RBC is not having mitochondria. Yes. RBC is absence of mitochondria. So can it take energy from beta oxidation? Beta oxidation, where it is happening? Mitochondria. It is not having mitochondria. So the beta oxidation of fatty acid will not happen in mitochondria. Ketone body synthesis is also not going to happen. So what is the energy source during fed state or fasting state or starvation state? It is only glucose. For RBC, it is only glucose. That's why if it is hypoglycemic, the first thing to get lysed is RBCs. Okay. Heart. In heart is more fond of fatty acids. Comparing to glucose, it will take fatty acid as its first energy source. But when we are in starvation, both are not available. Glucose is also not available, beta oxidation also not available. So during very severe starvation, but heart has to function. At that time, it will take ketone bodies as the energy source. Okay, for RBC, only glucose. For heart, the first preference is fatty acid, and during starvation, ketone bodies. What about brain? The first energy source for brain is glucose. It will not take fatty acid. It does not require beta oxidation of fatty acid. It will directly take ketone body as its energy source. For liver, during fed state, it is glucose. During fasting state, it is beta oxidation of fatty acid. During starvation, it is ketone bodies. So your liver, kidneys, other organs will be like this. When we have food, it will take glucose. Next step, it will take fat. Next step, it will take ketone body. But the exception is this three. Heart is more fond of fatty acid and ketone bodies. Brain is more fond of glucose than ketone bodies. Fatty acid is skipped in brain. And RBC, always only glucose. Okay. So now you know what is the importance of ketone bodies in our body. So that's why our body is storing more fat. Because when it need, it will give us either beta oxidation as fat or that can be converted to ketone bodies also. Okay, so ketone body synthesis. So where it is going to synthesis? In which organs? In all organs? No. Ketone body synthesis happens only in liver. All other things will happen in other organs also. This is a very important question. Which is the pathway happen only in liver is ketone body synthesis. Similarly, urea cycle also will happen only in liver. That when we are reading protein metabolism, we will see ketone body synthesis happens in liver. Okay. So it is happening in liver, but it cannot utilize that ketone bodies because of absence of the enzyme thiophorase. Absence of enzyme thiophorase. So what is the substrate for ketone bodies? The substrate for ketone body is also acetyl-CoA. The substrate is also acetyl-CoA. So here you can see acetyl-CoA is the substrate for ketone bodies. So what is the product of ketone body synthesis? Obviously, it is ketone body. What all those ketone bodies? This can be classified into primary and secondary. What is the primary ketone body? Acetoacetate. Acetoacetate is primary ketone body. And what is the secondary ketone body is acetone and beta hydroxybutyrate. This is secondary ketone bodies. Why this is primary, why this is secondary is during ketone body synthesis, acetoacetate is formed first. So it is primary. Beta hydroxybutyrate and acetone will be formed next. That's why it is second. Okay. So you know what is the substrate? Acetyl CoA. So now this acetyl CoA, not one acetyl CoA, three acetyl CoA. This is one acetyl CoA. This is acetoacetyl CoA, means two acetyl CoA. Acetyl CoA plus acetyl CoA is two acetyl CoA. Here you have two acetyl CoA. And here it is one more acetyl CoA. So totally three acetyl CoA will be combined to form HMG CoA. Clear? So how many acetyl CoA? You know substrate is acetyl CoA, but how many acetyl CoA is needed in ketone body synthesis? Means it is 3 acetyl CoA is needed in ketone body synthesis to form HMG CoA. So now this HMG CoA, it has 3 acetyl CoA. Yes, it has 3 acetyl CoA. Now this will remove one acetyl CoA and form acetoacetate. It is having 2 acetyl CoA. This is your primary ketone body. Acetyl CoA form acetoacetate. This acetoacetate again it will form one more acetyl CoA to form HMG CoA. Now this HMG CoA will lyse one acetyl CoA and form again acetoacetate. That is our primary ketone body, and then it will form acetone and beta hydroxybutyrate. So now you can ask why this has to happen like this. Why it has to form HMG CoA and why it has to again cleave. Already it is only 2. It can directly get the ketone body, yes or no? Why it has to form to this HMG CoA form and then again it has to cleave? You have that doubt? Okay. I will tell you. Before that, the important rate limiting enzyme in this step is HMG CoA synthase. HMG CoA synthase is the rate limiting enzyme for ketone body synthesis. Acetyl CoA forms acetoacetyl CoA. Again it will add one more acetyl CoA to form HMG CoA. So the enzyme involved here is the rate limiting enzyme for ketone body synthesis, that is HMG CoA synthase. Clear this step? So there is a significance of forming this HMG CoA. You know any idea what is the significance of HMG CoA? See, it has used 3 acetyl CoAs. So when it can happen? When we are having more fat. When we are having more fat, it will go to beta oxidation, but those intermediates and enzymes are depleted. It is diverted to this step. So in this step, we are utilizing 3 acetyl CoA to form an intermediate HMG CoA. Now this HMG CoA will decide whether body is in starvation state or body is in fed state. If we are in fed state, we are having more acetyl-CoA coming to this reaction because of more food. This HMG-CoA is diverted to cholesterol synthesis. So this HMG-CoA is an intermediate for cholesterol synthesis also. That's why it is forming HMG-CoA first. So if I am in fed state forming HMG-CoA, I won't synthesize ketone bodies. My body will synthesize more cholesterol. If I am in fasting state, now this HMG-CoA will be diverted to ketone body synthesis. That is, it is again cleaving it into acetoacetate, acetone, beta hydroxybutyrate. So now, who is going to take the responsibility of happening which reaction? The enzymes. For this, you know it is HMG-CoA synthase, that means synthesizing. Now if the enzyme is HMG-CoA reductase, HMG-CoA reductase, then it means it will go to cholesterol synthesis. If the enzyme is HMG-CoA lyase, then it will go to ketone body synthesis. Clear? Three enzymes with HMG-CoA. One is HMG-CoA synthase, and the another is HMG-CoA reductase, and the another is HMG-CoA lyase. You need three acetyl-CoA to form HMG-CoA. Okay, HMG-CoA has formed. Now the enzyme, which enzyme is coming into action, will take that HMG-CoA to that specific pathway. If lyase is coming, it will go for ketone body synthesis. If reductase is coming, it will go for cholesterol synthesis. So now who is going to do that function to decide which pathway to act? Tell me. Hormones. If insulin is more, it means our body is in fed state. Your insulin keeps HMG-CoA reductase in dephosphorylated state, and this enzyme also in dephosphorylated state. In dephosphorylated state, this is active, so it goes to cholesterol synthesis. When I am in fasting state, you know, my glucagon will be very high. My glucagon will keep both the enzymes in phosphorylated state. But this is the enzyme active in phosphorylated state, hence it will push the HMG-CoA towards synthesis of ketone bodies. Clear? Why it is happening like this? So your key HMG-CoA synthase is the rate limiting enzyme for ketone body synthesis. Your HMG-CoA reductase is the rate limiting enzyme for cholesterol synthesis. Yes? Okay. So what is this compound? All these are carbon. 1, 2, 3. The numberings are number of carbons. Okay. So finally you can see 27 carbons. Long chain fatty acid. This is not like a chain. It is like a ring. So it is not a chain fatty acid. So something 27 carbon fat. 27 carbon fat is called cholesterol. This is the structure of cholesterol. Cholesterol is made up of 27 carbon fats. The other name for cholesterol is cyclopentanoperhydrophenanthrin ring. Just see this term. They can, just for confusion, they can ask you cyclopentanoperhydrophenanthrin ring is made up of how many carbon atoms? That is nothing but your cholesterol, which is having how many carbon atoms? This cycle, I think they are mentioning this one, benzene ring. Like we have 3, and we have 1 pentone ring and 1 perhydro. And this is that phenanthrin ring, cyclopentanoperhydrophenanthrin ring. Yes. Okay. So you know this HMG CoA is from 3 acetyl CoAs. Yes, acetyl CoA with 1 more acetyl CoA forming HMG CoA. Now here the enzyme is HMG CoA reductase. Then what is this pathway? This is cholesterol synthesis pathway. So no need of all this enzyme, only this enzyme, which is the rate limiting enzyme, is important because statins inhibit this enzyme inhibiting cholesterol. Okay, just you have to know HMG CoA reductase. So this enzyme is activated by hormone insulin. So this enzyme will be active in which state? Dephosphorylated state. So finally, 1 by 1 carbon will be added to get 27 carbon cholesterol. Okay. So now you got cholesterol. Is cholesterol good or bad for health? Both. It has some functions also. It has some disadvantages also. A minimum amount of cholesterol is essential in our body for first is for absorption of fat soluble vitamins from our food. If we take fat soluble vitamin A, D, E and K, even if we take sufficient amount of vitamins, if my cholesterol is very low, our intestine cannot absorb those vitamins. So your cholesterol is like a carrier for those vitamins. If cholesterol is not there, it will go for excretion. So we need cholesterol for carrying fat soluble vitamins. It is essential for synthesizing steroidal hormones. It is essential for anything else? Fat soluble vitamins, steroids and bile salts. Bile salts, bile acid synthesis. So all this which is for routine function, we need cholesterol. Okay. So here you can see this is bile acid synthesis. What is the substrate for bile acid synthesis? Substrate, cholesterol. Your cholesterol is the substrate. Cholesterol is the substrate. Whenever it is a pathway, I will tell what is the rate limiting enzyme. The rate limiting enzyme of this pathway is 7-alpha hydroxylase. 7-alpha hydroxylase. So this bile acid synthesis will happen in liver. This bile acid synthesis will happen in liver. Half will happen in liver. The remaining half will happen in intestine. Because this enzyme 7-alpha hydroxylase is present in liver, so no other way it has to happen in liver. So once cholesterol in presence of 7-alpha hydroxylase, it will become alpha hydroxy cholesterol. It will form the primary bile acid. This is important. They can ask you very simple question. What is the primary bile acid? What are the secondary bile acids? So primary bile acid is cholic acid and quinodeoxycholic acid. So these two are primary bile acids. Why primary? It is forming first. So cholic acid and quinodeoxycholic acid. These two are the primary bile acids. Now this will get conjugated with glycine and taurine to form glycocholic acid, taurocholic acid, glycochinodeoxycholic acid or taurochinodeoxycholic acid. So primary bile acid is two. What are they? Quinodeoxycholic acid. If it conjugate with glycine, what is that? Only with glycine. Glycoquinodeoxycholic acid. If it conjugate with taurine, it is taurocholic acid or tauroquinodeoxycholic acid. So this is primary bile acid. So after this, from this liver, whatever has been synthesized reaches the gallbladder and enters to intestine. Once it enters the intestine, our intestinal bacteria will convert this primary bile acid into secondary bile acids. So what are those secondary bile acids? Deoxycholic acid and lithocholic acid, quinodeoxycholic acid and lithocholic acid. Yes. Done. So with this, fat metabolism is over. So now I will tell the metabolism and you have to tell me what is that rate limiting enzymes. First, I will start with fatty acid synthesis. For beta oxidation, ornithine palmitoyl transferase 1. Yes. So next, cholesterol synthesis. What is the rate limiting enzyme for cholesterol synthesis? HMG CoA reductase. So what is the rate limiting enzyme for ketone body synthesis? HMG CoA synthase. What is the rate limiting enzyme for bile acid synthesis? Yes. Okay. So how much of energy is utilized or synthesized in fatty acid synthesis? Utilized or generated? Utilized. How many? 2 or 2 or 1? Acetyl CoA carboxylase. It will convert ATP to ADP only. So it is 1. What about beta oxidation of fatty acid? How many utilized? 2 ATP utilized. How many produced? Depends on number of carbon atoms. Yes. So will cholesterol synthesis, I said any fatty acid, any ATP? No? No. In ketone body synthesis, is there any ATP production? No. In bile acid synthesis, did I say any fatty acids ATP production? No. Okay. So in ketone body synthesis, what is the primary ketone bodies? What is the name? Primary cholic acid. Is it ketone body? That's what. Don't confuse between ketone body and bile acid. Your acetoacetate is the ketone body. What is secondary? Acetone and beta hydroxy butyrate. Okay. So bile synthesis. What is primary bile acid? Cholic. Primary. One more. Cholic acid and okay. So what is secondary? Glycine and taurine are conjugated. Okay. That is conjugated primary bile acids. Secondary means what is happening in intestine. What? Okay. These are secondary bile acids. Clear? So what is this chylomicron? It is HDL. Apo A1 is seen in HDL. What is this chylomicron? Sorry, what is this lipoprotein? This is chylomicron. Okay. If B100 is there, it can be VLDL, which is out of this 4, which is carrying our intestinal fats. Chylomicrons, yes. Okay, which is having high protein content. HDL. Okay. What is mean by this beta lipoproteinemia? What is the defect in this beta lipoproteinemia? It is a hyperlipoproteinemia condition. It is type 3. What is the defect? Yes. So what are the clinical features you will expect in hyperlipoproteinemia? Tendon xanthoma. If it is tendon xanthoma, you should think about increase in LDL, okay, cholesterol. If it is Palmer or eruptive or tubuloeruptive xanthoma, then it means there is increase in triglyceride. This increased triglyceride, before coming as xanthoma, patient will have acute abdominal pain. Okay. So if it is hyperlipoproteinemia, the main clinical feature is xanthoma condition. So if baby, okay, whatever we have discussed from yesterday morning, I'm asking baby with hypoglycemia and hepatomegaly. If it is a condition like this, which one you will focus? Hypoglycemia with hepatomegaly means you have to galactosemia. Okay, before wait, I will tell galactosemia and fructosemia. I will tell you the clue. If it is storage disorder, inborn error, you have to think about liver glycogen storage disorders. In liver glycogen storage disorder, just they will give age of the baby with hypoglycemia and hepatosplenomegaly or renomegaly. If that same baby present with muscle weakness, exercise intolerance, then you have to think about muscle glycogen storage disorders. Okay. If you want to rule out galactosemia, the age of the baby will be between 1 to 6 months. Baby crying after feeding. Okay, baby crying after feeding. Along with it, you will have hypoglycemia, jaundice, in urine, reducing sugar, positive. So all this will point towards galactosemia. All these are same, just the age is more than 6 months. Then it means fructose, hereditary fructose intolerance. Clear? Your galactose and fructose are reducing sugars that can be excreted in urine, whereas you won't get any urinary finding in this glycogen storage disorder. So what else we discussed in inborn errors? Yes. Mucopolysaccharidosis. Yes. In mucopolysaccharidosis, what are the conditions we read? Do you remember? Hurlers, Hunters, Shees, and then Hurler, Hunters, Shees, Martex, Lamy condition. San Filippo is also there. So out of this 4, which is most severe form? Hunter, no? Hurler, yes. This is most severe. All are autosomal recessive except Hunter. And what is the main clinical feature for mucopolysaccharidosis? Mental retardation with corneal clouding. Anything else? All other common vomiting, feeding difficulty, failure to thrive, this and all you will see in everywhere. The clue is mental retardation. In glycogen storage disorder, no mental retardation. If it is baby with mental retardation, you have to think about either mucopolysaccharidosis or glycolipid storage, glycospingolipidosis. There also baby will present with mental retardation like Farber's, Gaucher's. In that condition also, baby presents with mental retardation. This corneal clouding is absent in Hunters. No corneal clouding. Okay. So can you tell me what is the enzyme deficiency in Hurlers? Alpha L iduronate or iduronate sulfatase? Okay. For this two, it is iduronate sulfatase. For Artex Lamy, it is aryl sulfatase. Okay, next glycospingolipidosis. Glycospingolipidosis, you know, GM3 gangliosidosis, GM2 gangliosidosis, GM1 gangliosidosis. Next, Fabry's Gauchers, Farber's, Niemann, Pick disease. Yes. So in all this condition, how about the baby? Mental retardation, cherry red spot with neurological manifestation and sometimes systemic manifestations. So what is the enzyme involved in GM3 gangliosidosis? Is it beta galactosidase? Correct. So this beta galactosidase will break galactose from this. Yes or no? So any other inborn error, have you seen galactose? Go to mucopolysaccharides. I said one thing which is having, instead of acidic sugar, it will have galactose. Is it keratin sulfate? Is it the keratin sulfate which has uraminic acid with galactose? So not uraminic acid, with the glucosamine with galactose. So have you seen galactose in GM3 gangliosidosis? Yes. And also in keratin sulfate? Yes. Check whether it is keratin sulfate or anything else. Mucopolysaccharide. Go to mucopolysaccharides. Chondroitin sulfate, dermaton sulfate, heparin, hyaluronic acid, keratin sulfate. Out of the 6, which is having galactose? Keratin sulfate. So your galactose is present in GM3 ganglioside, keratin sulfate, and also in myelin. What is myelin? Covering of our axonal nerves. Yes. So GM3, keratin sulfate, myelin, all are having galactose. Okay. For this galactose to break from any of this position, we need a common enzyme, beta galactosidase. So far I have said beta galactosidase in only one condition, that is GM3. But now I am going to introduce the role of beta galactosidase in other two, keratin sulfate and myelin. If patient is presenting with neurological manifestation, okay, if patient is presenting with neurological manifestation with mental retardation, then it is GM3 gangliosidosis due to deficiency of the enzyme beta galactosidase. If patient is presenting with corneal condition, then it is keratin sulfate due to that same enzyme. If patient is presenting with neurological manifestation with systematic manifestations, it is same enzyme affecting myelin. So that is called as Krabbe's disease. Krabbe's disease. So here you can see beta galactosidase causes GM3 gangliosidosis and also Krabbe's disease. Clear? GM, sorry. I'm so sorry. From down it is GM1. So first we will write GM3. With that we will add N-acetyl galactose, which will become GM2, and then it is galactose, which is GM1. So here it is GM1. Yes, yes. GM1 gangliosidosis. So this beta galactosidase is responsible for GM1 gangliosidosis and also Krabbe's disease. So in exam, your option can be beta galactosidase. Okay, your option can be beta galactosidase. So now you have to find the clue from the question whether it is mental retardation or neurological manifestation or corneal condition or with systematic manifestation. If patient is mentally retarded, obviously it is going to GM1 gangliosidosis. If eye problem is there, it is going to mucopolysaccharide keratin sulfate. If it is having neurological and systematic manifestation, it is pointing towards myelin degeneration, that is Krabbe's disease. Okay. So this enzyme is common for 3 things. So you have to differentiate depending on the clinical features of the patient. Clear? So with this, we are completing lipid metabolism. So 5 minutes, just have a look. In protein metabolism, there won't be ATP production. There won't be rate limiting enzyme. Everything is related to inborn errors. Only one enzyme for one pathway you are going to remember. Not like carbohydrate or lipid. It won't be a boring. It will be more interesting. Just one enzyme deficiency, this is going to cause this condition. Because from protein amino acid metabolism, we are not going to get energy. It is not going to help you during fasting or fed state. It is going to be involved in synthesis, protein synthesis, essential for DNA, RNA, and all other functions. If that is the absent, that protein is absent, what is the clinical condition? That's all. Okay. So I will give you 5 minutes. Just have a look. Then we will go to electron transport chain. We have completed lipid and fat. You know from where we are getting NADH, FADH. Now this NADH and FADH has to be converted to ATP. Okay, 5 minutes. Test will start. So the one pathway which is very, very important in our body is electron transport chain. So whatever maybe we are taking food or not taking food, we need energy for normal functions. That energy is in the form of ATP, will be given by electron transport chain. So what is the organelle where it happens? Mitochondria. Electron transport chain happens in mitochondria. In which organ it will happen is in all organs. Wherever mitochondria is present, ETC will take place. So the organ which is without mitochondria is RBC. Here, no ETC. So whether it happens in fed state or fasting state. Only in fed state. So in fasting state, if there is no electron transport chain, no ATP, so we will die. It's like that. That's why to prevent that, we have beta oxidation of fatty acid. So either in fed state or fasting state, we will get ATP, NADH, FADH. You know NADH, FADH from TCA cycle and beta oxidation that will enter electron transport chain for ATP synthesis. So it happens in both, either in fed state or fasting. ETC doesn't care whether we have taken food or not. It needs NADH and FADH to form ATP. It occurs in aerobic or anaerobic, both. Either in aerobic or anaerobic, we need NADH. So what are the electron transport chain components? So before writing, First understand what I'm telling, then we'll write only the exam related points. Yes, okay. So whatever in green are the components. For an electron transport chain, what is this? This is called, this is inner mitochondrial membrane. This is inner mitochondrial membrane. So this is mitochondrial matrix. This is intermembrane space. Okay. So your component of electron transport chain 1, 2, 3, 4 and 5. All these 5 are located on the membrane, not in the matrix. All 5 components are present on the inner mitochondrial membrane. This 2 is present on the inner side of the inner mitochondrial membrane. Correct? All are present as together, outer and inner combined, but the complex 2 is present in the inner side of the inner mitochondrial membrane. So these are the 5 components, 5 complex, which is required for final ATP synthesis. Okay. So now this is complex 1. So what is going to enter into electron transport chain is your NADH and FADH synthesized from various other carbohydrate and fat metabolic pathways. Whatever has been produced there, I said it will undergo oxidative phosphorylation. It has to undergo oxidative decarboxylation with phosphorylation. That is oxidative phosphorylation. What is meant by oxidative phosphorylation? What is meant by oxidation? With oxidation, what our body is going to get is heat. Yes, after oxidation, our body will get heat. It will gain more of ions, H plus ions. Only if oxidation is happening, we will get heat. Only if it is coupled with phosphorylation. Phosphorylation means adding of phosphate. We will get energy. That adding of phosphate is called, addition of phosphate to ADP is called phosphorylation. If this is happening in that cycle itself, I said it is substrate level phosphorylation. If it has to enter into electron transport chain, it is called oxidative phosphorylation. So if the term is oxidative phosphorylation is, we have various complex 1, 2, 3, 4. All these 4, what it is going to do is, it is going to release H plus ion to the space. So this here, it will produce more heat. That is not tolerable. So that has to again enter back to matrix to produce ATP. If only oxidation is happening, heat is generated, no energy is generated. Only if oxidative combines with phosphorylation, we will get ATP energy. Okay. So in complex 1, NADH will enter. From whatever the steps we have received NADH in the pathway, that NADH will enter through complex 1. Wherever we got FADH2, that will enter this electron transport chain from complex 2. Okay, NADH is from complex 1, FADH2 is from complex 2. So now this one, whatever the NADH has been entered, when NADH is moving through this chain, H plus will move out. Okay, so how many H plus moving out of complex 1 is 4 H plus. So when one NADH is moving from complex 1 through this chain, 4 H plus is released into space. Now this NADH will be transferred to coenzyme Q, which is just a carrier that will carry this electron from NADH to next complex. So from complex 2, FADH will give the electron to coenzyme Q, and this coenzyme Q is going to complex 3. So here there is no H plus. So only your complex 1 is giving 4 H plus, your complex 2 will not give H plus to intermembrane space. Okay. So now this has carried the electron to complex 3. Now in complex 3, when this electron is moving, it is also giving 4H plus. Yes, it is also giving 4H plus. So now this electron is moving to fourth complex. So this fourth complex is also giving 4H plus to intermembrane space. So finally, the electron has reached the mitochondrial matrix. Your oxygen with this formed water. When water has been formed, here it is 4, 4, here it will be actually 2. This is actually 2. Okay, not 4. This is actually only will get 2. So 4, 4 and 2 H plus has released to the space. Now your water has been formed inside. So now there is 10 H plus ion in the space, which is very heat, which is not tolerable. So now what is the role of complex 5 in electron transport chain is, it will take those which is in the intermembrane space to inside. It will come from outside to inside of the mitochondrial matrix. So once this H plus is coming in, 1 ATP is generated. So for 1 H plus, 1 ATP is generated? No. When 4 H plus are coming in, 1 ATP is generated. Clear? When 4 ATP is 4 H plus are coming in, you will get 1 ATP. Clear? Totally 5 complex. First complex, NADH is entering to transport chain. From second complex, FADH2 is entering. Your coenzyme Q and cytochrome C are just the electron carriers. They are not going to do anything. So this complex 1, 3 and 4 will give H plus to intermembrane space. H plus is not released from which of the following complex, means from complex 2. So totally 4 plus 2, 10 hydrogen ions entered into the space. When this 10 has to enter back into the matrix, ATP is generated at a rate of 4H plus entering 1 ATP. So can I write 4H plus when it is entering, it will give you 1 ATP. Clear? So now you can tell me when NADH is entering here, how many H plus is formed? When NADH from complex 1 is entering. So 4, 4, 2. Totally 10. So for 4 H plus, you know 1 ATP. So 1 plus here it is only 2. So 0.5. That's why 1 NADH is giving only 2.5 ATP. Why NADH is giving 2.5 ATP is, NADH is entering to complex 1. From 1, you will get totally 10 H plus. All 10 when it is entering back, 4, 4, 2. So it is 1 plus 0.5. You will get 2.5 ATPs. This is for NADH. That is the calculation why NADH is getting 2.5 ATP. Now FADH. FADH2 is entering in second complex. Yes. So from second complex, you have to calculate how many H plus has been released. Leave this first one, which is not involved in carrying FADH2. So when FADH2 is entering to complex 2, you will get no H plus from complex 2. But 3 will give 4. Complex 4 will give you 2. So 4 plus 6, 4 plus 2, 6. When 6 H plus are entering inside, 4 H plus will give you 1 ATP and the 2 H plus will give you 0.5 ATP. And that is the reason 1 FADH2 will give you 1.5 ATPs. Understood why it is 2.5 and 1.5 ATP for NADH and FADH? It depends on in which complex it is entering and how many ATP generated. Clear? So if only H plus is entering to intermembrane space, which cannot go back inside, what will happen? That means oxidation happen. H plus has entered to intermembrane space, oxidation happened. But this phosphorylation is not happen. Maybe any inhibitors or anything. It means heat is generated. Yes. So is there any physiological condition where we will get only oxidation without phosphorylation? So now I am going to make that electron transport chain in a circular way. This is my complex 1, complex 2. I should not make complex 2 like this. I will make like this, okay? Complex 3, my complex 4, and finally complex 5. So here it given 4H plus, this has given 4H plus, and this has given 2H plus. So totally I have 10H plus in my intermembrane space. So now this 10H plus has to enter through complex 5. Instead of this, there are some conditions where we will have a transporter, H plus transporter in the mitochondrial membrane. So this H plus, instead of moving through this complex, it will move through this transporter. So what will happen? Only if it goes through the complex 5, ATP can be synthesized. If this is not going to complex 5 and moving through that other transporter, H plus has been happened, oxidation has been happened, but no ATP production. So this is most commonly seen in brown adipose tissues. Brown adipose tissue. So this kind of a mitochondrial membrane is common in newborn babies. Because to keep the baby in warmth, our baby's body itself is having a condition of generating more H plus, oxidation happening with more heat. Instead of no need of much energy, no need of much ATP production. So this is happening physiologically in newborn babies, which is seen in which cell? In which cell mitochondria it is seen? Is not in all cells. In adipose tissue. Adipose tissue, adipocytes, mitochondria will have this transporter which make the baby to feel warm. Clear? So along with it, there are some conditions such as thermogenin and high thyroxine production. All these conditions, there is only oxidation, no phosphorylation. And similarly, if it is any drug, we can tell dinitrophenol, dinitroprazole. So these two are uncouplers. What is meant by uncoupler? Means oxidation with phosphorylation. When it is combined, it is coupling. Only oxidation is happening without phosphorylation, means it is called uncoupling. That is uncouplers. Dinitrophenol and dinitroprazole, if we are going to accidentally ingest, what it will do is, it will block this complex 5. So H plus will go out, oxidation will happen, but phosphorylation will not happen. That is called as uncouplers. So these are synthetic uncouplers, which is synthesized and given toxic. Whereas these conditions are physiological uncouplers happening naturally. Clear? Okay. So you understood what is happening in electron transport chain. So now start writing. Complex 1. So for any complex, for any reaction, we need an enzyme. So for complex 1, the enzyme, what is that is going to transfer is NADH. So the enzyme is, it is going to transfer NADH to coenzyme Q. Oxidation is happening with reduction. So it is NADH, COQ, coenzyme Q, oxidoreductase. Clear? Complex 2, it is going to carry FADH. But what is that? That is going to give this FADH is succinate. In TCA cycle, succinate. So the enzyme is succinate, coenzyme Q, oxidoreductase. If you find instead of succinate FADH, it is also right. It is complex 2. What is complex 3? So what is this complex 3? In complex 3, not like NADH, FADH, only this coenzyme is going to carry to cytochrome C. So it is Q cytochrome C oxidoreductase. Your coenzyme Q and cytochrome C are just the carriers of electrons. So what is in complex 4 is, what is here in complex 4? So here the cytochrome C is getting oxidized. So it is cytochrome C oxidase. So these are the enzymes present in electron transport chain. If any of this enzyme is absent, defective, electron transport chain will not happen. NADH-CoQ, succinate CoQ, Q cytochrome C and cytochrome C. So now here, what are those 5 protein complex means? Complex 1, complex 2, complex 3, complex 4 and complex 5. This 5 is having other name, that is ATP synthase complex. It is going to synthesize ATP, so it is called as ATP synthase complex. What is the one non-protein in the complex means? It is that coenzyme Q. It is only carrier. There is one peripheral membrane protein, that is cytochrome C. So from this complex 1, it releases 4H plus. In complex 2, no H plus is released. Complex 3 will release 4H plus and complex 4 will release 2H plus to intermembrane space. Yes. So I said about 1, 2, 3, 4. So what is complex 5? You know complex 5, the other name is ATP synthase complex. So shall I make like this? Okay. So this is complex 1, 2, 3 and 4. This has released H plus. So now comes that final complex, which is ATP synthase complex. It is having 2 subunit, F0 subunit and F1 subunit. F0 is present on the membrane and F1 subunit, which is responsible for that ATP synthesis. Conversion of ADP plus PA to ATP will happen in this F1 subunit. So when 4H plus entering from F0 to F1, this part will rotate, not F0. F0 will not rotate. This F1 component will rotate. So when it is rotating, ADP can combine with PA to produce ATP. So this F1 component which is rotating will generate ATP. When it will rotate is when 4H plus enters inside, it will rotate and give 1 ATP. Okay. So you know what all that 5 complex, how many H plus is released? Where NADH is entering, complex 1. Where FADH is entering, complex 2. Since NADH is entering in first complex, how many ATP it is producing? 2.5. Since FADH is entering in second complex, it is giving only 1.5 ATP. So that number depends on number of H plus released into space, intermembrane space. So this is normal. Uncouplers, there can be some physiological condition and also synthetic condition. In physiological condition, for newborn baby, they will have more of brown adipose tissue. Our body will have white adipose tissue that will not have that transporter. Brown adipose tissue will be having more of that natural transporter of the H plus. So that helps the body to keep it in warm state. So it is either due to increased brown adipose tissue, increased thermogenin, or increase the thyroxine levels can cause that uncoupling. Uncoupling means oxidation is happening, ATP synthesis, phosphorylation will not happen. So if it is drug, it is 2 drugs, dinitrophenol, dinitroprazole, which is responsible for that same uncoupling. Okay. So now comes the inhibitor. So there can be many inhibitors which can inhibit at different complex. Clear? I think last July also they have asked one question in this. It is regarding complex 3 they have asked. So we'll see from one by one what all the inhibitors inhibiting which complex. So the first complex inhibited by amobarbital, pyrazidine A. Amobarbital and pyrazidine A. Complex 2 is inhibited by the compound malonate carboxyl. Complex 3 is inhibited by the compound antimycin and British anti-Leucid ball. Complex 4 is inhibited by the compounds carbon monoxide, hydrogen sulfide, sodium azide, cyanide. This is for 4 complex. Then that 5th complex. In 5th complex, we have 2 parts, F0 and F1. F0 is inhibited by oligomycin. F0 is inhibited by oligomycin and F1 is inhibited by atractyloside. Oligomycin atractyloside. In complex 5, ATP synthase complex, oligomycin is inhibiting F0. It means oligomycin prevents entry of H plus into complex 5. Clear? Oligomycin prevents entry of H plus into complex 5. What atractyloside will do is, atractyloside will allow H plus, but it will not allow phosphorylation. It will not allow conversion of ADP to ATP. So they can ask you, conversion of ADP to ATP is inhibited by not oligomycin, it is atractyloside. Which of the following prevents the entry of H plus ion into complex 5? It is oligomycin. So barbiturates drug inhibit which of the following complex? Barbiturates drug inhibit which of the following complex? Complex 1. And then malonate carboxyn, these are present in some compounds. Better you go and check malonate and carboxyn or component of any toxic poisons. Okay. In first complex, amobarbital, pyrazidine, and one more is rotenone. So this rotenone is, I think, rat poison. So in rat poison, if we ingest rat poison, death is confirmed because it is inhibiting electron transport chain complex 1. So if rat poisoning patient, it is very difficult to handle. They surely will go for death because of this inhibition. So similarly, find out what about malonate and carboxyn, whether it is present in any common insecticide something. Okay. And complex 3, it is antimycin and BAL, British anti-levisate, that will inhibit complex 3. And then the gas, hydrogen sulfide, carbon monoxide, cyanide poisoning, and sodium azide poisoning. All this will inhibit complex 4. And finally, oligomycin and atractyloside will inhibit complex 5. Clear? So this is about inhibitors. So you should know the components, what are the enzymes, and what are the inhibitors. So can you write this 1, 2, 3, and 4? What is that enzyme? What is for complex 1? What is the component is, you know, for 1 it is NADH, for 2 it is FADH, succinate, for 3 it is cytochrome B and C, for 4 it is cytochrome A and A3. Here it is NADHCOQ, oxidoreductase. For 2 it is succinate COQ, oxidoreductase. For 3 it is this. If you have written already, write the uncouplers. What are the 2 drugs? Synthetic 1, which is uncoupler. What about the physiological uncouplers? Present in brown adipose tissue. What is present in brown adipose tissue is dermogenin. Dermogenin is the transporter which is present in brown adipose tissue. And then thyroxine, and also long chain fatty acid can also be a physiological coupler. So now you write this inhibitor, complex 1. Tell me what is for complex 1? Rotenone. Okay, complex 2. Okay, malonate. Okay. What is complex 3? Antimycin. BAL. Okay. For 4. Sodium azide. Okay. ADP to ATP conversion. It is inhibited by conversion. Atractyloside. ADP, ATP translocase by stopping entry of H plus, that is oligomycin. Okay. Okay. That's all with electron transport chain. So each and every word can be an MCQ in electron transport chain. Anything they can ask you. Uncouplers, inhibitors. Inhibitors are most common questions. Okay, just take 2 minutes, go through that inhibitors. Yes, close your note. Close your note. Carbon monoxide inhibits which complex? Complex 4. Antimycin inhibits 2 or 3. Yes, carboxyl inhibits 2. Malonate inhibits. Okay. Amobarbital. Barbiturates inhibit complex 1. Sodium acid inhibits. Oligomycin inhibits. 5. Atractyloside inhibits. Complex 5. So how much amount of ATP is generated from NADH? 2.5. How many H plus it will release? How many H plus will be released from NADH? 10. How many H plus is released into intermembrane space by FADH2? ATP is 1.5. How many H plus? 6. 4 in third complex, 2 in fourth complex. So totally 6. Okay. So what is physiological uncouplers? Name some physiological uncouplers. In a brown adipose tissue, it is dermogenin, thyroxine, long chain fatty acids. Drugs, DNP. Okay, what is the expression? Dinitrophenol, dinitrocresol. Dinitrophenol, dinitrocresol. Okay. So what is the carrier, electron carrier in electron transport chain? Non-protein electron carrier. Cytochrome C is protein. Non-protein is coenzyme Q. Okay. So we'll start with amino acids and proteins. Okay. So name is amino acid. So it means it is going to have both amino group and acidic group. Clear? So if I make one carbon, I will make acid on my right side and amino group on my left side with one hydrogen on top and one aliphatic group at the bottom. So this is the structure of amino acid. That's why the name is amino acid. Amino on left, acid on right, hence it is called amino acid. It is one H and one aliphatic group. Anything can join there. So if two amino acid is going to combine, will it looks like this? This is one amino acid. Now my another amino acid. Can I write like this? NH2, H, NH, yes. Can I write like this? This is NH2 only, yes. So this is my amino acid 1 and this is my amino acid 2. When my two amino acid has to join, I need a bond. And this bond which is going to be present between my two amino acid is called peptide bond. What is the bond seen between two carbohydrates? Glycosidic bond. So the bond which is present between two amino acid is peptide bond. So will it be like this? No, the peptide bond cannot form like this. It will remove one higher water group. It will remove that water group. So to form a peptide bond, it has removed this water group. So now the bond is between CO and NH. So this is how my peptide will form a bond. For each and every amino acid to join, one peptide bond will combine these two with removing one water molecule. So this is called peptide bond. So peptide bond is between CO and NH. So shall I write like this? Yes. So can you tell me how many amino acids is here? This is one peptide bond. Okay, how many peptide bond? This is my one peptide bond. Peptide bond is between CO and NH. No other doubt. These are not peptide bonds. That is the amino acid structure. Whatever the bond which is between CO and NH is that peptide bond. So I have three peptide bonds here, which is between CO of one amino acid and NH of another amino acid. Okay. Okay. So if I mention the same structure, amino acid, this is the structure I said. What is mean by asymmetric carbon atom? Asymmetric carbon atom means the carbon atom where its 4 valencies are joined by 4 different groups. 4 valencies which is having 4 different groups. Then it is called asymmetric. If all 4 is, here it is H, here it is acid, here it is amino group, here it is anything. So 4 valencies having 4 different groups are called asymmetric carbon atom. There is an exception which is not an asymmetric carbon atom is the simplest amino acid, which is NH2CCOOH. Here it is H and here is also H. So this is the structure of an amino acid called glycine. So in glycine, you can see this carbon having 2 same group. So your glycine does not have asymmetric carbon atom. There is no asymmetric carbon atom in glycine. Yes, all other amino acids will have asymmetric carbon atom. One will join CH3 group. One amino acid will form CH3, CH2 group. Other amino acid can form CH3, CH2, CH2 like that. So the one which is not having asymmetric carbon atom is glycine because the both positions are only hydrogen. Okay. So how we are going to get amino acids? It is synthesized in body or we have to take as food? Both. Some amino acids can be synthesized in our body, but some amino acid we have to take it. Depending on that, we can classify the amino acid as essential, non-essential. Essential means essentially you have to take it in food. It is very essential to take it in food. That is called essential amino acid. Those which if we are not going to take in food also no problem. Our body will synthesize itself. They are classified as non-essential amino acids. So totally how many amino acids we have for a function? So why we need amino acid? Amino acid is for protein synthesis. Peptide bonds will join to form a long polypeptide chain. That polypeptide chain, see like this peptide bond will join to form a long polypeptide chain. This polypeptide chain will get folded to form active protein. This is also protein. Polypeptide chain is also protein, but this is not active. When it is in straight line as a chain, it is not active. Only when it is folded into tertiary structure, it will become active protein. What is mean by active protein? It will be involved in any functions. So to involve a protein in any function, we need that protein to be inactive. To keep that protein inactive, it should be present in its it should be present in 3-dimensional folded structure. A linear polypeptide chain cannot do its function. Clear? So they can ask you whether protein will be active in polypeptide chain, secondary structure, tertiary structure, or quaternary structure. So this is actually called the level of organization of proteins. At first, this is polypeptide chain. Now this will get folded to form secondary. Now this will get folded to form secondary structure, and now this will further gets folded to form tertiary structure, and finally we have one more thing that is called quaternary structure. So here I have added 2 colors. It means your quaternary is made up of 2 polypeptide chains. Your primary, secondary and tertiary is made up of a single polypeptide chain. Only the changes is in folding. In quaternary, it is made up of 2 polypeptide chains. Clear? So out of this 4, the active form is this one and this one. All the proteins will be active in tertiary structure. There are some proteins which is active in quaternary structure. The example is hemoglobin. In globin, you know, we have alpha globin and beta globin. So we have alpha polypeptide chain and beta polypeptide chain. So some proteins which is made up of two polypeptide chain has to present in quaternary structure. Proteins which is made up of a single polypeptide chain has to get folded to be active in tertiary structure. For example, insulin. Yes. Not insulin actually. Yes, insulin only. At first it will be two A chain and B chain, and finally it will be joined by disulfide bonds. Okay. Where I started? Okay, asymmetric. I said about essential and non-essential. Okay, how many amino acids are there? 20 amino acids for a normal function. Now we have discovered two more amino acids. One is called selenocysteine and the 21st, and the another one is called pyrrolysine. Okay, how this amino acid is converted to protein? What is the mechanism? What is the machinery involved in synthesis of protein from amino acid? What is the machinery involved in synthesis of protein? You know translation? You know replication, transcription, translation? Yes. So translation is the technique, molecular machinery which is present in our body for adding up of one by one the amino acid to form a linear polypeptide chain. That is our final proteins. This we will study molecular biology tomorrow. Okay, protein translation, how amino acids are converted to proteins. Okay, so what is mean by derived amino acids? Derived amino acids means there are some amino acids, glycine, serine. Yes, can I classify like that? If I classify an amino acid, I can classify it as simple amino acids, glycine, alanine. I can classify it as acidic amino acids, that is aspartic acid, glutamic acid. To classify it as basic amino acid, not basic, amide amino acid, asparagine, glutamine. To classify as sulfur containing amino acid, cysteine, methionine. All these are cysteine, methionine. Cysteine is from cysteine only. So all these are sulfur containing amino acids. To make us an aromatic amino acid, aromatic means ring structure. Ring structure will be present in phenylalanine, tyrosine, tryptophan. To classify the amino acid as branched chain amino acid, what all will come under branched chain amino acid is leucine, isoleucine, valine. There is one amino acid, that is proline. So like this, we can classify the amino acid depending on the property. If it is having acid, if it is having sulfur, if it is having ring structure, and if it is having branched chain. So all these are normal amino acids. Then what is mean by derived amino acid? Derived amino acid is something derived from this normal amino acid. For example, proline, if it undergoes post-translational modification, that is hydroxylation, it is called as hydroxyproline. And also lysine, there is one more amino acid, lysine, which is hydroxylysine. So like this, if it undergoes either hydroxylation or glycosylation, means addition of glucose unit to an amino acid. So either hydroxylation, addition of hydroxyl group to an amino acid is called hydroxylation. Addition of glucose group to an amino acid is called glycosylation. Addition of a phosphorus group to an amino acid is called phosphorylation. So all this kind of post-translational modification, if an amino acid undergo, then those amino acids are called as derived amino acids. So where this derived amino acids found? It can be found in protein also, it can be found in not found in proteins also. So what is the most important derived amino acid found in protein? Means the first thing should come to your mind is collagen. You know collagen is a protein, fibrous protein, which is having hydroxyproline and hydroxylysine. So derived amino acid which is found in protein is collagen having hydroxyproline and hydroxylysine. Hydroxyproline and hydroxylysine. So the one which is not found in protein is ornithine. Ornithine is also a derived amino acid from glutamine. This is not found in protein. Where ornithine is present? It is present in urea cycle. Ornithine, citrulline will come under urea cycle. So this is not found in protein. If you are not remembering this, no problem. Just remember this two which is found in collagen. This is very important when it comes to collagen, I will tell you. Each and every third amino acid is made up of hydroxyproline and hydroxylysine. That is the importance. Okay. Okay. So next. So here in this picture you can see the center one Zwitter ion, also called as isoelectric pH. So can you see here the charge is getting neutralized, 1 plus and 1 minus, so it will get neutralized. So in isoelectric pH, it is the pH where your amino acid has no charge. What is mean by isoelectric pH? Isoelectric pH is the pH of the medium, the solution where the amino acid is there. So it is the pH of the medium where amino acid is having no charge. So this charge is getting nullified. So here the charge is 0. When charge is 0, as I said in electrophoresis, it will move depending on the charge or mass. If charge is getting 0, it cannot move. So here migration is absent. It cannot migrate. It will stop when it reaches the charge of 0. So consider if this amino acid is going in an electrophoresis from cathode to anode, this amino acid is moving in this way. At some pH in that medium where its own PI, isoelectric point, has been received, after that it will not move. It will stay at that point. That is called as isoelectric pH, where your migration will be absent. It will stop. So it is the point where it will get maximum precipitability. It will get maximum precipitability and minimum solubility. It will get precipitated at that point. It cannot move. So this is the properties of isoelectric pH. What is mean by isoelectric pH means the charge of that amino acid is equal to the charge of that pH of that medium. This is Pi, the amino acid's charge. When Pi, the charge of an amino acid, is meeting the pH of that medium, both are in equal condition, then that is the point of isoelectric point. Okay, so now you can see high pH and low pH. What is happening here? High pH and low pH. So now I will make a trough which is filled with high pH. High pH means what? pH is more than my amino acid's isoelectric point. So this is the medium where my pH is high. pH is high means H plus will be less. Yes, pH high means H plus. What is pH? 1 by H plus, log of H plus. So here pH is high. pH is high means it is alkaline. It is an alkaline condition. In alkaline condition, we will have less H plus. So in this medium, if I am going to put my amino acid, okay, if I am going to put my amino acid, which is having only less H plus, it cannot come and bind to my amino acid. Clear? Here my amino acid cannot attach this H plus because already H plus is very less. So in this medium, my amino acid will exhibit negative charge. Clear? Why it is negative charges? Only in that solution, if it has H plus, amino acid which is negatively charged can take that H plus and form a H plus positive condition. Already H plus is less in the medium, so it cannot take that H plus. So your amino acid exhibit negative charge. I will give one more example. Consider pH is less than Pi. What is mean by pH less than Pi? Acidic. Acidic condition. When pH is less, it means acidic. Acidic means we have more H plus. Yes, the medium will have more of H plus ion. That's why pH is less. In this condition, if I put my amino acid, all this H plus will come and attach on the surface of my amino acid. So now my amino acid will exhibit positive charge. Clear? That's why in low pH, it will exhibit positive charge because more of H plus. When more H plus is there, it will attach here and attach here. So this will become neutralized and NH3 will be positive. But in condition like high pH, already there is less H plus. So what my amino acid is doing, it is giving one H plus to the surface and now it is becoming negatively charged. Understood? If it is surface is having less pH, it is giving the H plus ion. If surface is having more pH, it means alkaline. If surface is pH, which is alkaline, alkaline means how about the H plus? H plus is less. When H plus is less, my amino acid is giving one H plus to the surface and now it is becoming negatively charged. In condition like low pH, acidic condition, the surface is having more H plus. Now my amino acid will take my H plus and it will become positively charged. Clear? So this is what is happening to my amino acid depending on the medium. That's why albumin will be positively charged or negatively charged depending on that solution. In HCl, in our stomach and intestine, when albumin is taken, it will exhibit positively charged. But in our blood, albumin will exist in negatively charged. It depends on the medium in which it is present. Clear? So the important thing is you should know all this property of isoelectric pH. So just you can write this classification of amino acid. You can't remember everything. So what I will prefer is read only that essential. So all other remaining things are non-essential. Along with essential and non-essential, we can classify it as glucogenic or ketogenic. What is meant by glucogenic or ketogenic? When amino acid is going for catabolism, whether it is going to give you glucose or it is going to give you ketone bodies. Anything related to TCA cycle intermediates or gluconeogenesis intermediate, those amino acids are taken as glucogenic. Anything which is going to give you acetoacetate beta hydroxybutyrate, those amino acids are taken as ketogenic. Clear? For example, succinate, fumarate, malate, those are TCA cycle intermediates that can go for gluconeogenesis. So those are glucogenic amino acid. If it is something going to give you acetoacetate or acetone, that is called ketogenic amino acids. So this group is both essential and also glucogenic. This group is both essential and ketogenic. This group is essential with both glucogenic and also ketogenic. In this, the most favorable question is leucine and lysine. They will ask you leucine and lysine whether it is ketogenic or glucogenic or both. And they can ask you one non-essential amino acid which is both glucogenic and ketogenic. One non-essential amino acid which is both glucogenic and ketogenic, tyrosine. This is leucine. This is leucine and lysine. Yes, done. So now we'll go for metabolism. So when it comes to amino acid, one vitamin which is very, very important is pyridoxine, vitamin B6. Not only here, anywhere in further metabolism, you will see vitamin B6 often in amino acid metabolism. Okay. So here, the first one is transamination and oxidative deamination. So what is this function? Why we need transamination and oxidative deamination to happen in our body is wherever the amino group ammonia which is getting deposited, that has to go back to liver. Why? If some ammonia is accumulating in brain, if some ammonia is accumulating in blood, that has to go back to liver because urea cycle happens only in liver. The function of urea cycle is excretion of ammonia, removing of ammonia from our body. So the urea cycle happens only in liver. So our body has to collect all ammonia from all the tissues and take it to liver. So the first one happening for this is transamination. So transamination means transferring of amino group. Okay, transamination means transferring of amino group, any amino acid with one keto acid getting transaminated. It means this amino group is shifted to this keto acid. So now here you can see glutamate. It is an amino acid and you will get one keto acid. So now I will write one example here. Alanine. Is it an amino acid? Is alanine an amino acid? Yes. Okay. Along with alpha keto glutarate, which is a keto acid. Now this alanine is going to give the amino group to alpha ketoglutarate to form an amino acid which is called as glutamate. So when this is going to give the amino acid here, what you are going to get here is one keto acid which is your pyruvate. You know pyruvate won't have amino group. So this is transamination. The enzyme involved here is alanine transaminase. The enzyme involved here is alanine transaminase. Similarly, aspartate with alpha ketoglutarate, it will form pyruvate and glutamate. So here the enzyme involved is AST, aspartate transaminase. So this is going to happen in all the tissues, all organs. Finally, we got glutamate. Yes, finally we got with amino group glutamate. So now this glutamate from whatever may be the tissue will be transported to liver. Till this step it is called transamination, happened anywhere. Now it has to go back to liver. Why it has to go back to liver is it has to give that ammonia only to liver. So transamination happened. Now this glutamate is coming to the liver. In liver, glutamate will be dehydrogenized. That is NADH is getting dehydrogenized to form back alpha ketoglutarate and ammonia is getting free. Clear? So this enzyme is present in our liver, which is glutamate dehydrogenase. We don't have alpha ketoglutarate dehydrogenase for this action. We don't have alanine dehydrogenase, anything like that. We have only glutamate dehydrogenase. Since we have only glutamate dehydrogenase, whatever may be the amino acid, it has to convert to glutamate because we have glutamate dehydrogenase. So only whatever the amino acid, if it converts to glutamate, it comes to liver. This enzyme will act on glutamate to give alpha ketoglutarate and give this ammonia into liver, which will enter urea cycle for excretion. So transamination happens in all the cells, all organs. Oxidative deamination, this is called as ammonia is getting freed, that is deaminated. So this oxidative deamination happens only in liver. So this is transamination and oxidative deamination. This is very important because transamination requires vitamin B6. Transamination, all transamination reaction requires vitamin B6. If this is not happening, if this is glutamate transferring to liver, if transamination is happening, oxidative deamination is not happening. So if I take our brain, in brain we have glutamate. Okay, now this has to go back to liver for removing that ammonia. If it is not happening, this will be converted to glutamine in the brain itself. Glutamate will be converted to glutamine by the enzyme glutamine synthetase. So this glutamine is having that osmotic effect. It will absorb water to the brain, causing cerebral edema. Clear? So this should not happen. Only to prevent this to happen, this has to go back to liver. Clear? So to clear the ammonia from brain, we need glutamate. So to remove ammonia from muscle, what do we need? Consider skeletal muscle. In skeletal muscle, this is liver. This is liver. In liver, glycolysis has happened. Pyruvate, and this pyruvate will goes to muscle, and this will be converted to what? During severe exercise, your pyruvate will convert to lactate. But during fasting, your pyruvate will convert to what? Alanine. Yes. So whatever alanine is an amino acid having ammonia group. So this alanine during fasting is accumulated in muscle. So this alanine should not get accumulated in muscle. So it has to come back to liver, and this alanine is one of your substrate for gluconeogenesis. Clear? So this cycle is called as glucose alanine cycle or Cahill cycle. One is called Cori's cycle, that is for lactate. That is during exercise. But during fasting, alanine is accumulating in muscle. Nowhere ammonia should accumulate. To remove that ammonia from the muscle, alanine will come back to liver and it will enter to gluconeogenesis. Similarly, in brain, glutamate is there. It should not be converted to glutamine. To prevent that cerebral edema, glutamate will come back to liver and undergo oxidative deamination. Clear? So this is that functions. So here actually the glucose pyruvate or glucose will come. Alanine gluconeogenesis will be pyruvate and this will be liver. So this will go back. This is for removing ammonia or clearing of ammonia from muscle. So which amino acid is involved in this work is alanine in muscle, glutamate in brain. Okay. In brain also, see this is blood-brain barrier. Okay, blood-brain barrier, NH3. Once it is entering into the brain, will convert this glutamate to glutamine. This glutamine is having osmotic effect, so it can absorb water, causing edema in brain. And what about this glutamate? Have you heard of GABA? Inhibitory neurotransmitter, gamma aminobutyric acid. So this glutamate can form GABA, which is an inhibitory neurotransmitter which will get accumulated in brain. So all neurological functions will be affected. Hence this has to cleared off from the brain. Okay. So next comes glycine metabolism. Glycine can be I'm not going to explain this mechanism and all, not at all needed. So glycine can be synthesized from serine, threonine, which are other amino acids. Serine, threonine can form glycine, and this glycine can be converted to glyoxylate with help of the enzyme glyoxylate aminotransferase. It is a reversible reaction. This step is important. Glycine, reversible reaction, it forms glyoxylate. And what is the enzyme involved here is glyoxylate aminotransferase. Clear? So this glyoxylate, if it is not returning back to glycine, it will get accumulated. If this enzyme is not functioning, it will get accumulated as oxalate. This oxalate can be excreted in urine. That condition is called hyperoxaluria. So glyoxylate aminotransferase. You know, if it is aminotransferase, obviously your vitamin B6 has a role. So either deficiency of this enzyme, that is this enzyme, glyoxylate aminotransferase, or deficiency of vitamin B6 leads to accumulation of glyoxylate, oxalate, which is excreting in urine, which is called as hyperoxaluria. So they can ask you what is the enzyme deficient in this condition. Glyoxylate aminotransferase, or which of the following vitamin deficiency can cause hyperoxaluria? It is vitamin B6. So any of these two can cause. If it is primary hyperoxaluria, it is that enzyme deficiency. If it is vitamin B6 deficiency, it is called as secondary hyperoxaluria. Glycinuria means transporter defect. There is defect in transporting glycine to the intestine for absorption. So transporter defect, whatever taken in food or synthesized cannot go back to function. It will be excreting in urine. What is non-ketotic hyperglycinemia means? It is the defect in glycine cleavage. Once the function is over, it has to get cleaved, degraded. So if it is not getting cleaved, degraded, it will start to accumulate. Glycine accumulation. Glycine is an amino acid. So along with that amino acid, ammonia group is also getting accumulated. So it will cause us encephalopathy, which is toxic to brain. So it causes encephalopathy. Why it is encephalopathy is glycine, which is an amino acid having an amino group, which is toxic to brain, causing encephalopathy, that is non-ketotic hyperglycinemia, defect in cleavage system, glycine cleavage system. So why this is important is all this 4 disorders of glycine metabolism is important. Yes. So and one more important part in glycine is this glycine has role in creatinine metabolism, creatinine synthesis. You know what is creatinine? Synthesis of creatinine. It is for which test? Creatinine is for renal function test. We will see serum creatinine, urine creatinine. Yes. So your glycine is a substrate for creatinine metabolism and also for heme synthesis. In this too, your substrate is glycine. So here you can see this is synthesis of creatinine. No need of any pathway. Only thing you should know is what all the amino acids involved in synthesis of creatinine. What all the amino acid involved in synthesis of creatinine? Arginine, glycine. That's all. Only glycine and arginine. Can you see anything? What all the amino acid involved in synthesis of creatinine? Glycine, guanidine. No, guanidine is not an amino acid. Methylation. Methylation means wherever there is a methylation, methyl group is given. So who is going to give that methyl group is methionine. Yes, adenosyl methionine, which is the active form. This is the third amino acid, that is methionine, which is a sulfur containing amino acid, which is going to give methyl group. So the third amino acid which is involved in creatinine synthesis is methionine. They can ask you all of the following amino acids are involved in creatinine metabolism except arginine, alanine, glycine, methionine. So the answer is alanine. Arginine and glycine will form guanidoacetic acid. This, after getting methylated, it will form creatine that will go phosphorylation to form phosphocreatinine, and finally phosphate will be released and creatinine will be synthesized. Only these three things you should know for creatinine metabolism. So why in exercising muscle lactate is produced? Why in fasting muscle alanine is formed? Yes. Will there be lactate available? For conversion of pyruvate to lactate, you need NADH. We are already depleted in fasting condition. We don't have much energy to provide NADH and all. So what your pyruvate has to do? It has to take up ammonia and form only alanine. This cycle is called, what is this? Cahill's cycle. You can see alanine is transferred, that gives the ammonia, and this glucose is circulating. So you should not confuse between Cahill's and Cory's cycle. Cory's is lactate. This is alanine. So next comes catabolism of branch chain amino acid. Don't write everything. Just you can write this step. You should know what all the branch chain amino acids. They are leucine, isoleucine, valine. These three are branched chain amino acids. I will make it here leucine, isoleucine, valine. So this first it will undergo transamination. First it will undergo transamination, and then it will undergo the important oxidative decarboxylation. Oxidative decarboxylation. And finally, it will give different products that I will come later. So why this is important is the defect in oxidative decarboxylation. Okay. The defect in this oxidative decarboxylation, the enzyme involved here is branched chain keto acid dehydrogenase. It is branched chain. They are branched chain amino acids. So it is branched chain keto acid dehydrogenase. When this enzyme, this is not a single enzyme. This is a multi-subunit enzyme. It has many other subunits. So when this enzyme is absent, oxidative decarboxylation will not occur. So what is this condition called? This condition is called as maple syrup urine disease. Maple syrup urine disease due to oxidative decarboxylation defect, the enzyme deficiencies, branched chain keto acid dehydrogenase, which is a multi-subunit enzyme. So what is mean by maple syrup is patient will be having burnt sugar odor in urine. When this step is not going to occur, whatever may be the keto acid formed before step will be excreted in urine. So that is going to give burnt sugar odor in urine. Along with that, patient will be having vomiting, lethargy, not able to take food, feeding difficulties, everything. Because amino acids which we are going to take from food is not getting metabolized. Okay. So your clue will be in question. Obviously, they will mention you burnt sugar odor. If it is burnt sugar odor, it is maple syrup urine disease. So what is that investigation we will do for this test is, this is the test. Can you see some yellow color fluid? When you add urine with dinitrophenylhydrazine, it will form some yellow deposits. It means urine is having branched chain amino acid which is getting deposited. This test is called DNPH test, done for babies with branched chain amino acid metabolism defect, especially maple syrup urine disease. Maple syrup urine disease. DNPH test. They can ask you what is that biochemical test you will do for MSUD babies. DNPH test. Yes. So now I will come here to this metabolism. You know, valine, isoleucine, leucine or branched chain. What is that enzyme defect causing maple syrup urine disease? You know. So now see at the last, if metabolism is going to happen, your valine will give propionyl-CoA. Your isoleucine is giving acetyl-CoA and propionyl-CoA. Your leucine is giving acetyl-CoA and acetoacetate. So in this, propionyl-CoA will be going to glucogenic fate or ketogenic. Propionyl-CoA can convert to succinyl-CoA, succinate, TCA cycle, gluconeogenesis. So this is having glucogenic fate. That's the reason you have written before the classification. Some amino acid as glucogenic and some amino acid as like ketogenic and some as both. So now here you see propionyl-CoA is glucogenic. What about acetyl-CoA? Acetyl-CoA is also entering into TCA cycle. It will give oxaloacetate, gluconeogenesis. So this is also glucogenic. Then what about leucine? Acetyl-CoA is glucogenic. What is acetoacetate? It is a ketone bodies. Yes, so it will have ketogenic diet. So now check that page whether you have added leucine under both glucogenic and ketogenic. You have added leucine under both glucogenic or ketogenic. No, in this structure they have given leucine only in ketogenic. This is actually partially glucogenic and partially ketogenic. Why I am telling this is, in exam, if they ask you which of the following is ketogenic and the options are leucine, lysine, any of this, methionine, histidine, then the first option and the best option is lysine. Your lysine is the first option to call it as purely ketogenic. If lysine is not there in your option, then only you can go for leucine as an answer. Because leucine, you have seen, it is both glucogenic and ketogenic. It has partial glucogenic action activity also. So the one thing which is only ketogenic is lysine. Okay. Second option only you have to think about leucine. When both are there, don't think about leucine. So this is branch chain amino acid. You should know what all the branch chain amino acid. You should know what is maple syrup urine disease, what is that enzyme deficiency, and what is the investigation test, and what is that key clinical feature for maple syrup urine disease.